Morning. So we'll start with the previous lecture, that is novel ingredient technologies for food application. So what we covered last time or previously was all about biorefinery and about why do we need new technologies and how we can introduce new technology into different bioprocessing application. So as we discussed the other day, so we'll so far whatever we have covered, if you have any questions, well, you should have at least one or two questions. So we'll start from that corner.
Sir, a day before yesterday you you told us about integrated biorefinery concept for microalgae.
Yeah.
In which you discussed about 4 steps: cultivation, harvest, cell disruption and extraction.
Yeah.
Cell disruption and cultivation. So there are 4 processes in which there are different combined, means in which first all 4 are in different stages. In second method, cultivation and harvest are together and other than. Sir, can you tell which one of these 4 processes are more efficient and more better or
okay.
To do so?
Now as far as different combinations were given, it depending upon the condition. Say for example, if you have continuous wastewater coming or continuous water is there and microalgae is growing, so there you can, the cultivation is going on, and wherever the cells are forming in larger quantity and they can be separated, and that can be disrupted, that means by using ultrasonic or any other technology, and then you can carry out your extraction process. That is cultivation, separation and disruption together, A. B, in some cases when you have a specific bioreactor to produce a specific species, so you do the cultivation, clear the bioreactor, harvest your cell, that's harvesting separated, yeah, and then you dry it for further processing, that's another application. Third one is you have cultivation, then you do harvesting, disruption together, because when you harvest it, it's still in a liquid form, you don't have to dry it for further extraction. You can do the extraction in wet biomass itself, either by fermentation or by using enzyme. So, and then if you go for centrifugation, that is separation process, you can segregate your lipids, proteins and whatever bioactive you are doing it. So depending upon your core objective, whether you are growing microalgae for phytoremediation purposes, then you can have all 3 things together, because there will be a continuous flow of wastewater coming into the stream. So it will be depend upon what is your target biomass and target ingredients, food application, feed application, or only for phytoremediation. Accordingly you can select the concept. Yeah.
Sir, as we do harvest and cell disruption together, does it affect the final product or?
Well, each and every unit operation will definitely affect, because when you are doing harvesting, that is collection, for example, if you have a continuous water flow system, yeah, where you have all the microalgae cells are growing along with the water, and then you have a filtration system here, yeah, and that means all the cells are trapped here, and then whatever water moves here and then it is circulated, and in this case all the concentrated cells are collected here and then you process it. So that all combinations again depends upon how efficient you want the system to be. That means if you are doing harvesting, cultivation, harvesting and disruption all together, so there will be changes in the system, has to be configured or designed accordingly. The principles remains the same. The purpose of showing those 5 different combination was, since biomass is not like duckweed or a seaweed which has to be separated, so it makes things easier. Yes, sir.
Good morning, sir.
Yes,
so my
good
question
morning.
is about bitterness of spirulina.
Bitterness of
Spirulina. As we noticeably when it is fresh or in dried, which time is more bitter, and any other treatment for reduction of bitterness, and can bitterness affect on nutritional quality of spirulina, sir?
Okay. Now, as far as bitterness is concerned, bitterness is not only in spirulina, in majority of the protein there are some issues or the other. Some certain type of bitterness is because when you create a peptides, that is due to enzymatic processes, yeah. Debittering enzymes are there, or flavorzyme is FLAVORZYME. Flavorzymes are the one which can capture those amino acid or bind or encapsulate those amino acid which leads to which leads to bitterness, A. B, bitterness is also because of the quality of the protein what we are getting. So if you see the milk protein, they have a specific taste. If you go for fish protein, they have a specific smell. Every protein is having different, even when you go for pea protein or soya protein, they do have a beanie flavor. Yeah. So every protein is not only, although protein is tasteless, but whatever other compounds are attached with that, if you do identification of those compounds, and accordingly mitigation strategies can be adopted. So one of the most commonly used is flavorzyme, and that happens when we do protein hydroslate. Debittering bitterness happens because of glutamic acid, aspartic acid, certain combination of amino acids, or the peptide, which is where having a terminal amino acids, all those which causes bitterness.
I have another question.
Yeah.
Currently, I am working on banana blossoms. So when I will take it for processing, after cutting it takes immediately brown, turns brown. I have applied different types of browning techniques like KMS or sodium benzoate or alum treatment, oxalic acid, lime water, but I have not got proper results. So any other techniques for
It's browning of what biomass?
blossoms, sir?
Badam? What is that?
Banana blossom.
Oh, banana, okay. No, the browning is just because of, again, as we discussed about polyphenol oxidase and phenolic.
Yes.
So if you try for the blanching application, that should be able to sort out that browning problem. Because you use KMS and all are there for anti-browning agent, but the moment you are cutting it and if you are leaving it for longer, if it automatically turns brown, then it becomes very difficult. It's a time lapse because enzyme acts very fast. You slice a potato, straight away goes for blanching, it won't turn brown. You slice the banana, slice the potatoes, leave it for half an hour, then you go for hot water blanching, it won't solve the problem because phenolics are already converted into quinones. Maybe just the time lapse between that when it's harvested, when it is landing to your place, and then subsequent treatment when you are doing it. So that has to be added straight away while doing so. So one way is before you slicing and cutting it, if you crush that one in, if you add directly whole bit into hot water and make an emulsion or make colloidal suspension of that in that particular hot water itself, that should sort out the problem. And even if it turns brown, but your objective is to get the proteins out of it, is it? What is the main function of that? What is your end product?
So extruded product is final product is extruded product from
Just
that,
to make a powder.
product, sir. Extruded product. Hot extrusion.
Okay, hot extrusion. So even though if you when you inactivate microorganism, and you also have to see whether that browning is because of your enzymatic browning or non-enzymatic browning. That particular banana shoot might have lot of sugars, and some bit of protein might be causing non-enzymatic browning, that is Maillard reaction products. Yeah.
Good morning, sir. So my question is, like as our water bodies are getting polluted, so they might cause some allergic reaction to human body, right? So what could be those allergens in fish, and can they be removed?
Okay, so seafood allergy is a major concern, right? It's all either it's because of shellfish allergen, yeah. So shellfish allergen, whether seaweed, duckweed or anything, every person is having different sensitivity. Some they have allergy against a specific type of protein. Allergies can be removed by, first one is all these are protein. Say for example there is a seaweed and there are small shellfish attached to it, yeah. So different interventions, for example even drying reduces the allergenicity. Complete elimination of allergies or allergic compound is quite challenging, but you can reduce that one. And certain allergies are also associated with shells as well as for fish, pavin, pelvin and all these fish allergens. Some are inherent, so people have to stay away from those ones. As you see, like it may contain allergens. So interventions are all related to the technology. Some of these technologies what we discussed, that reduces the allergenicity, and bringing allergenicity to zero is quite not possible, but reduction in allergenicity is possible. Whether it's a peanut allergy or it is related to your shellfish or because of soya bean, all these are basically a protein which reacts differently for different person's body, yeah. When it comes to duckweed, seaweed or microalgae, there are certain shellfish which grows in the water, can attach to the biomass, But the moment you go for cleaning process, going for drying processes, and those proteins are not inherent of macroalgae or microalgae, so that can be easily degraded or can be denatured. Now I am using that word because that particular protein is denatured, then allergenicity can be reduced.
Good morning, sir. My question is, what are the explain the extraction process of omega-3 fatty acid in fish oil?
Omega-3 fatty acid from where?
Docosa, hexanoic acid and
No, that's right. From which source?
fish
Biomass.
oil. Fish oil.
Fish oil. Now, when we use fish, if you do fish meal, yeah, that means if you cook whole fish into hot water condition, 80 degrees Celsius for 30 minutes, so you get a fish paste, yeah, and then you separate lipids, yeah. Once you get a lipids, and lipid, you get oil, phospholipids and everything. So if once you have oil, which is a structure of glycerol plus fatty acid, then you have to use an enzymes to break those lipids into or that's oil or fat into the specific fatty acid, and then you have to separate it by using any other chromatographic technique to separate specific fatty acids. That's the only way, and that largely depends upon what biomass we are using. That applies for almost all the oils, yeah. Whereas when it comes to grain, you have to get the lipids out. Grains, because they have low moisture content, the processing strategy will be different. Take for example wheat germ oil, yeah, wheat germ, and then you have oil. Oil is very small quantity, so you may have to use either supercritical CO2 extraction, you get your oil, and then this can be fractionated or broken down by using an enzyme, that is lipases, yeah, and they can separate your glycerol with the fatty acid, and then that can be fractionated by using chromatographic techniques.
Thank you.
Sir, shall I ask my question at last?
That's okay.
Thank you, sir.
Sir, Clostridium sporogenes cannot be destroyed at a high temperature. Even if we try to kill it by increasing the temperature, the other microbes also got affected. Then what's the alternative way to kill it, sir, without affecting the
Which
other
one?
microbes?
Clostridium.
Ah.
So Clostridium can be affected at temperature, but temperature and time combination is so high that it will impact other nutrients. Yeah. So we have to come up with a combination where we are not using very high temperature. Food safety is one thing. Even if you inactivate Clostridium botulinum, that can be inactivated at 121 degrees Celsius or certain time and a pressure 15 psi. But in order to retain the nutritive value of the product, we have to come up with an alternative technology. That means we demonstrated or we discussed yesterday about high pressure processing to germinate those spores and then to use a temperature. Yeah. So the combination of thermal along with non-thermal technique can solve that problem. Again, the question remains, even if we inactivate the microorganisms, how we retain its nutritive value. Yeah. Because in model solution, it's easier to inactivate microorganisms, but when it comes to the food matrices, they provide that protective effect. Yeah.
Good morning, sir. Sir, my question is from the biorefinery session. So, what type of cavitation method of ultrasonics are used in the processing of whelk shells? So basically there are two types, right, sir? Direct ultrasonics and indirect. So what type of cavitation method is used, and for what kind of food materials it is used?
Okay. You store your questions. So when we cover today, we are going to go through the ultrasonics, and that will answer your question. If it doesn't answer your question, then you can ask that particular question. Don't forget that one. Yeah.
Okay.
We have plasma next, and after that we are going to go through ultrasonics. Yeah. So far you have seen only the application part of it. Bit of fundamental and basics has not been covered yet. Yeah.
Okay, sir. Thank you, sir.
So, good morning, sir.
Good morning.
So, my question on plant protein isolation, sir. We are while doing isoelectric precipitation method, we are using hexane, HCl and NaOH. So, once I have done freeze drying, still there is any chemical residue left behind in the powder sample. So, if it is there, what are the impact in food while incorporating?
Okay, any of the chemical which is used, there will be always some odor residues will be there. Take an example, even if you wash your clothes and there will be always odor. Even if you remove the smell and odor, two different things, right? And hexane residues will be there because hexane what you obtain is not ultra pure. Yeah. Same thing for hydrochloric acid, whichever is used, there are laboratory reagent, analytical reagent, and depending upon the purity. So there certain residues are always left over. If it is ultra pure, it can be removed, but at the same time, the purpose of removal of hexane or ethanol from the production line is from health and safety perspective. When you scale up the processes, then you have to make lot of provisions for health and safety, for explosivity, for health and safety of the person operating on it. These are the main concerns of replacing hexane. So best one is not to use hexane and to come up with alternative solvent. Yeah. So there you might be compromising with the yield, but at the same time at least you have a safer product. Yeah.
Good morning, sir. My question is that almost all plant-based ingredients has anti-nutritional factors. These anti-nutritional factors like tannin, saponin, and phytate, these are the anti-nutritional factors. Like how to reduce the anti-nutritional factors same ways without compromising the nutritional content, because some anti-nutrition factors are like heat sensitive and some are heat stable. For heat sensitive, we can give heat treatment to reduce the anti-nutritional factors, but same ways we are getting compromised in nutritional factors also. So how to overcome this thing?
Okay. First of all, they are not anti-nutritional, now we call them a non-nutrients. Yeah? Nutrients, and there is difference between the components which provide the nutrition and other one they have a pharmaceutical benefit. So vitamins are not nutrients. Yeah? Protein, fat and all, they are nutrient which provides energy and different functions, whereas vitamins are considered as as cofactor enzyme or vitamins which provides support to the system. Now you mentioned tannins, phytic acid, saponins. Yeah. Now in tea there is a epicatechin, gallicatechin. Yeah. They also are tannins. Yeah. So they are not harmful. The only problem is when they are present, they bind with certain nutrients which makes them non-available. Yeah. Like when we say when you drink green tea is perfect, when you take green tea with the milk proteins are bound with these tannins, and neither the benefit of tannin nor the protein can be observed. Yeah. So the way to remove it is, again, if they are attached with the protein, there is a specific then the reason is some of those treatment can eliminate those non-nutrients. Other one is if you have to separate it, then you have to see the structure, how the tannin is bound to which type of protein and what a specialized enzyme to be used to break that particular bond. So they are not present in it, they are bound with certain types of protein. Yeah. So that is one, and at the same time saponins, phytic acid, these all are also beneficial for some other compounds, some other processes. So that can be separated again by using a specialized solvents or coming back with depuratic solvent. So coming with a robust way of doing it, because when we deal with the biological matrix, so it's not that it will be separated just like separating mixture of different type of coins. Yeah.
So then, so why duckweed in food application is less in India compared to Western countries?
Not even Western country duckweed is used much, but in commercial production is only at the moment in Israel and America. Yeah. Whereas certain part in Southeast Asia duckweed is cultivated and consumed. Yeah. Now that is to do with the socio-political, and all those factors. Yeah. Even though we have large amount of seafood seashores, majority of population is not non-vegetarian. Yeah. Even though we have animal resources, still majority of population is vegetarian. So these are social, cultural aspect, and when people they see it's having some benefit, then consumption starts. Yeah. Like 30 years ago, 30 years ago, nobody was knowing about pizza, burger and all those things. Everything was samosa and chai. Yeah. Now samosa has been replaced by croissant, burger, and that means again how we are interacting with the international community and how people are promoting specific type of food product. That is one thing, and slowly it starts, and depending upon we can propose what is there best for the nutrition. It is up to the individual to select what they want to eat. Yeah. Even if you know that chole bhatura is having too much of oil, but still people they eat it. Yeah. So that's the one of the reason for social aspect and cultural aspect of consumption of those biomasses.
Sir, one more question is that how to increase the phytochemical profile of the plant samples? Like many treatments were equipped to increase the phytochemical profile, but the thing is, after treatment, it will be increased in the amount, or else it will break down the cell wall and increase the concentration of the phytochemicals.
Okay, so what I understand your question is how you can increase the bioactives or phytochemical yield in your extract, yeah? Okay, take any biomass, name the biomass, anything. Oh. Take it, say for example tea, yeah? So you want to have an extract which contains higher amount of bioactives, yeah? That is phenolics. So in tea, when we start extraction of the process, if you add a water, you always get some extract, yeah? And there are lot of lot of tea extracts are sold, yeah? Like ready to drink tea extract for phytochemical. The moment you add a water, in that water you will get a phenolics, that's why color is changed, plus you will have whatever is soluble in the water. You will have a soluble fiber, yeah? You will have soluble proteins and all other large molecular weight compound. And if this liquid goes through molecular weight cutoff, yeah, of 10 kilodalton or 5 kilodalton, so less than 10 kilodalton membrane will have all those compounds which are lower molecular weight. So you can concentrate the phenolics there, yeah. One is the way you extract it, you get lot of bioactives into liquid suspension, yeah. Other day when I was mentioning, even though extract yield is increased, yeah, whereas the content yield is reduced, but it reduced just because what all has been solubilized in that particular water suspension, yeah.
Good morning, sir.
Yeah, go on, yeah. Good morning.
My question is about in biorefinery process in India. As per 2023 schedule, Indian seafood are mostly wasted above the 30 tons. In order to reduce the wastage, what are the ways to...
Only 30 ton in India?
30 million tons.
Okay, yeah.
30 million tons in India. So this is due to the heavy metals present in Indian nearby oceans. In order to reduce, what are the methods, techniques, like combination of high pressure and high temperature, any other method to use?
No, it's actually when it comes for see heavy metal contamination, that's a bigger issue not only for seafood, almost every product. Pesticide residue, they also contains organopesticides, metallopesticides, they all are getting into the food systems, yeah. Now again, for removal of heavy metals from side stream is the same issue what is there from the mainstream, yeah. So best will be again to do the extraction processes and come up with the way how we can segregate the heavy metals, yeah. Either by, if water is there, most of the heavy metals they are not attached with the protein and the free bound, if they are in a suspension or in ash content, that again with activated carbon sand filter, that's the way heavy metals are currently reduced. Cannot be eliminated, that is there. Again, the important thing here is where and how these type of heavy metals are incorporated or present in the particular biomass, yeah. So it will be number of different strategies has to be adopted. So I won't have a clear answer how we can remove heavy metals. If I do have a clear answer, we'll solve the world's problem of removing heavy metal from the food matrices.
So my another question is in milk industry, in the production of casein protein, compared to whey protein, the casein protein is lesser amount in productions. During the casein productions, we use the rennet as a protease. In order to increase the casein productions, what are the other protease except than rennet, which is more cost efficiency? What are the other methods to increase the casein production in milk?
Now it's actually when it comes to milk protein, you have a casein and whey protein. Whey protein is also basically a casein, isn't it?
Yeah.
Yeah. So that means you are asking how we can increase the casein in whey protein. When we say whey protein, that means protein coming into whey. Yeah.
Yeah.
Milk protein broadly is classified as casein. Yeah. So if it is coming the milk protein concentrate. Yeah. or milk protein isolate. These are name of the biomasses, and the protein coming from whey, it will be whey protein isolate. But in all the cases it will be casein, isn't it?
Yes, sir. Sir, after then using rennet mostly in milk industry, other any method to production of
rennet is used for breaking the emulsion. So now lot of acids are used for separation of protein. You know, like rennet is used for coagulation purposes. Yeah.
Yes.
And then you also use lime, tartaric acid, what is used domestically. These are also coagulating agents, you know, to coagulate the milk protein. So that rennet is one of those. Then you have acid. You also have lot of even alcohol. The different chemicals are there which will just disturb the isoelectric point of the protein, and it will remove the protein.
Yes. Sir, when we add in the rennet in the extraction of milk casein, the other nutrients does not affect.
Rennet is an enzyme, right?
Yes.
Yeah. So that's other nutrient in terms of what reduced.
Milk contains a lot of calcium as a nutrients.
Well, when you are removing the protein, see, calcium is there, calcium, vitamins and different ingredients are there.
Yes.
But again, it's the purpose, if you have a protein, the nutrients has to be separated anyway, you know.
Yes.
And some nutrients will come with the protein, some won't come in.
Yes.
So that depends upon whether you are interested only in separation of vitamin or separation of proteins. If you want to make 95%, 90% of protein, there won't be chance of whatever nutrients, other nutrients are coming in, that will be considered as an impurity. But getting 100% protein, that means you have to eliminate all other nutrients. Yeah.
Yes.
And remember, when we are talking about alternative protein, all other bioactives, vitamins and other things which are coming along with the protein is also important. Especially those who are dealing with alternative meat product, the problem lies in how we complement vitamin B12, what is that? Cyanocobalamin. Yeah.
Yes.
That is only animal origin product. So certain amino acid, or sorry, certain vitamins has to be there along with the protein. Then only we consider the nutritive value of that particular protein. Yeah.
Okay. Thank you, sir.
So good morning, sir. So why freeze concentration cannot be used to remove microbes in fish?
Sorry.
Why freeze concentration cannot be used in remove microbes in fish?
Fish concentration
cannot be used
for
freeze concentration.
Fish protein concentration. No, I didn't understand.
Freeze concentration cannot be used in remove microbes in fish, sir.
Fish concentration cannot be used for, I didn't get
freeze.
the question.
Freeze concentration.
FIS is fish.
Freeze, sir.
Sorry, I didn't get it. Sorry.
Freeze.
FISH. Freeze drying. Oh, sorry. Yeah, freeze drying cannot be used for
remove microbes in fish.
Microbes. No, when you do freeze drying, it will dry the microorganisms also. Yeah. And freeze drying is one of the method also to dry the microbiological culture for different product application. See, when you try to buy a lactobacillus culture, yeah, some of those microbiological cultures are freeze dried. Yeast, which is sold in the market for baking, is also instant freeze dried yeast culture. Yeah, it will only dry the microorganisms. The moment it gets the water or its conducive condition, it starts growing. Was that your question? How we can use freeze drying for removing the microorganisms? Yeah? No?
Good morning, sir. Sir, my question is related to from the extraction of protein from crab cells and from those things. So we are getting as a solid biomass called chitin, which you mentioned we can use it as a biofertilizer. So my question is that, sir, whether
sit down. Yeah.
We can use that chitin as a biofertilizer to increase agricultural production. So if it is possible, means we can use this chitin and we can achieve that sustainable practices, which it will not affect environment and it promotes soil growth as well. So whether it contains same that nitrogen, so nitrogen, phosphorus and potassium which we are using as a fertilizer in agriculture production, whether we can use it as an alternative. Is there any practice carried in any part of the world in agricultural production, so that our Indian farmers also can adopt this practice?
Okay, now when it comes to, if you, I'll try to answer your question from the agronomy side of it. There are two elements here, right? One is amount of fertilizer required for a crops to grow. Current specification for wheat or cereal crop is 180 nitrogen, yeah, 60 NPK, yeah, plus other nutrients are required, yeah. For 180 kg nitrogen, you use around 360 kg of urea, yeah. But if you look into the practices, you don't need that much amount of fertilizer for plants to grow, yeah. That means if 180 kg of nitrogen is used and all the nitrogen is converted into protein, and see the percentage of protein in biomass and amount of conversion. So that is, plant is having net nitrogen utilization, I think NNU, yeah. Amount of nitrogen utilized by plant is, I think, 30 to 40%, right? And since this is a concentrated form of fertilizer, part of that is absorbed by the water. If you know, if we have a plant, you keep flooding it with the water. Do you think what plant need that much amount of water? No. So drip irrigation reduces the water by significantly, I don't know exact percentage, by almost like by 80 to 90% water can be reduced, right? Same way, fertilizer content requirement is less for the plant compared to chemicals which are used, right? Now coming back to biofertilizer from chitin, chitosan, they contains all the nutrients and different minerals which are required for plants to grow, yeah. And if that is added to the soil, the leaching of those nitrogenous fertilizer will be limited. That will retain in the soil, plus it will also build up the microbiome of the soil, which will enrich the soil health, yeah. Because if you are using a chemical that is standalone, is not a part of the soil, and it is easier to run off, yeah. So that means whatever is replaced, that's why organic cultivation is quite expensive compared to chemical one, because at the moment chemicals are cheaper, right? Now when it comes to where these practices are, half of the European countries, they already have a target to replace agriculture by 50% through organic cultivation. Then again it comes to your glass house cultivation for a specific type of vegetables, specific type of crops. They also need a fertilizer, and the chitin or chitosan plays an important role. They also act as an antifungal, yeah. So one side is the cereal crops, whereas when you talk about vegetable crops, tomatoes and all those vegetables, they need number of different nutrients like calcium, mineral, magnesium and all those are present in those shells, which can be used in combination with the seaweed. Seaweed is also used as a soil conditioner because of presence of alginate. So there are a lot of success stories are there where farmers are already using this. Even those who have a poultry farm, they use poultry litter for fertilizer purposes. Cow dung, that's again left nitrogen and all, is also used for fertilizer. So that's the way it has to be. First, we need to know the requirement of the plant. Based on that, fertilizers are formulated. Yeah.
Thank you, sir.
Good morning, sir. My question is, during the shrimp shell biorefinery process, does it affect the yield of chitosan if we use different species of shrimp shells in biorefinery steps like deproteinization, demineralization, etc.?
Yeah, it's again whatever we are obtaining, recovery yield, purification and extract yield largely depends upon how much is there in a biomass. Certain species, the whole purpose here was shrimps, at the moment time is not there where shrimps are grown only for chitosan purposes. That means if you starve shrimps and give more calcium rich diet, so they will develop only chitin or chitosan on the surfaces, less of muscles. It's something like, if you have a big apple, but may not have that much of nutrients, just the moisture there. In a similar fashion, depending upon the species and variety, whatever is and how much is their composition, and that will dictate how much chitin, chitosan we will obtain. Yeah. Same thing about your soft crab. Other day you were asking and you mentioned afterwards that there are certain varieties, they don't have hard shells. So that's again the varietal differences. So like a blue crab, they have different chitin level, size of the crab. That's a big, I've seen even the crab which has almost like 2 pound, that is around half kg or 700 grams, big massive crabs. I've also seen crabs which are only 50 grams or 60 grams. Yeah. So that's all depending upon how much is the original composition or what is the chitin composition of original shrimp.
Thank you, sir.
Good morning, sir.
Yeah.
Ethanol is used to remove water content from the hydrocolloids at this precipitation occurs. Other than ethanol, which alternatives we can use to precipitate hydrocolloids?
Precipitate
colloids.
Hydrocolloids. One is alcohol, that means removal of water, that's one, right? Other one we discuss about the membranes. Yeah. Hydrocolloids are large molecular weight compounds, whereas impurities will be sugars and low molecular weight compound. So using the membrane technique, we can separate those hydrocolloids. Membrane is the simplest replacement. Apart from that, that's the only way we can replace ethanol as of now, or that is what I know at the moment. Yeah.
Thank you, sir.
Good morning, sir. My question is, what are the limitations of using integrated multi-trophic aquaculture in the production of microalgae?
IMTA for microalgae?
Yes, sir.
Not for macroalgae?
Macroalgae, sir. Sorry. Macro.
No, IMTA, see, basically when we are using aquaculture system, so either you are growing shellfish, finfish, mussels and all, yeah, and then they produces lot of residues as well, like feces, unburnt food and other things, yeah. So if we are doing aquaculture and along with that if we have seaweeds growing, so that means the concept was to have all the aquaculture system together, yeah. Something like there is agriculture and a forestry. Now the promotion was there for agroforestry, tree around the field and the crops there, yeah. So that are the different types of systems coming in. Or again there are advantages and disadvantages. Some places you can only have seaweed cultivation or without having any the shellfish, finfish cultivation, whereas some places you want to make same aquaculture system for harvesting and all the infrastructure. If you have integrated system, so you can have multiple crops, it's in the same time duration. When I say crop means same lot of biomass can be produced. Any other question? Yeah.
No, sir. Thank you.
Good morning, sir. Sir, can you suggest some example for the natural green solvents for the extraction of essential oils from the sea products?
That was my own question. I was asking you to think about and tell me what can be the thing which can replace hexane. Yeah.
Yes.
So that's a $1 million question. If you do find some natural green solvent, yeah, there are certain solvents which are claimed to be greener compared to hexane. That's as I mentioned about other day. TMO, no, not TMO, it's actually one of the solvent which is having higher boiling point compared to hexane, but they do leave the residues into the sample, right. What I suggested other time alternative was to do the co-extraction with other vegetable oil, yeah. So if you are removing, even membranes are quite are okay with the polar solvent, not with non-polar solvent, yeah. That is, membrane is also having its own limitation to remove oil. Is by using essential oil plus vegetable oil. If you take any other vegetable oil which can be used as a co-extraction purposes, or solvent free extraction by using a physical technique, or supercritical fluid. So there are not much alternatives there.
Thank you, sir.
Telepathy. Yeah, go on, yeah.
Sir, in duckweed, the water-soluble protein is Rubisco. Is that Rubisco have any functional properties? I mean, the amino acid compositions, any bioactive compounds?
Yes, there are a lot of almost this is an aquatic plant, yeah. Like any plant, that also contains a lot of phytochemicals, yeah. That's having your starch, Rubisco, and Rubisco is also having some functionality, and if you want to introduce a functionality to it, again you have to go for peptide processes, B, and it contains a lot of bioactive phytochemicals which has already proven to have antimicrobial activity, bactericidal properties as well as functions for biopesticides, yeah, A. and then it also contains a lot of vitamins and also a good source of vitamin B12 because it is coming from aquatic region. It does contain those, and that since it's a new biomass has been getting explored for different purposes. Even though we know tea phytochemicals from last 100 years, but still we keep looking into different functions. Yeah. So as of now, what the knowledge is, it contains phytochemical, they do have antimicrobial activity. Once we know if extract is having antimicrobial activity, then to purify that extract, that is which exact compound is causing, and then to come up with the point how much is the content, and then if you are to improve that particular bioactive, then you can change the production processes for duckweed. Yeah, it does contain.
Hello sir. So while we were discussing about the effects between different novel technologies, like between HPP and coal plasma and other things, we were discussing about the oxidation of it. So in coal plasma, it majorly works on the principle of oxidation and oxidative reactive species. So what about this oxidative stability of the product that has been treated with coal plasma?
Okay, now this is actually one, we all know that if anything is treated, yeah, it won't be as a natural, yeah, there will be oxidation happening. So there if you see what is, I'll ask a question and it will answer your question, right. Now, if suppose vitamin oxidation, yeah, I'll take it this way, you are worried about vitamin oxidation, yeah, if you are using with the plasma for any product or any bioactive, and it contains E. coli, yeah. So what will be the D value to reduce 90% microbial population of E. coli, yeah, condition required to achieve 90% reduction in E. coli? Will that be higher or 90% reduction in oxidation of vitamin will be required? Yeah. E. coli can be inactivated by using plasma in 1 minute. Yeah. Whereas if you give 1 minute treatment to vitamin solution, you can get only 5 to 10% reduction. Yeah. That means D value of chemical compound is way too high compared to microorganisms. Yeah. So the conditions required to achieve the food safety objective, yeah, will reduce the vitamin and oxidative product, but not to the level what is caused by what is required for E. coli. Yeah. A. B, when we are replacing thermal, so in a similar condition, if thermal is having the similar effect of plasma, there is no point in replacing it. Yeah. So the whole purpose of replacing thermal was because thermal will have a larger impact on vitamins compared to oxidative one. Another point is these vitamins and all other bioactives are present inside the food matrix. Majority of the plasma treatment is surface decontamination. The microorganisms are not inherent part of the food system. They come from outside sources. So they are attached. So attachments are easy to remove rather than which is present internally. So if you want to oxidize the vitamin within a plant using plasma, that will be difficult to do it. Yeah. Whereas removal of microorganisms, killing of microorganisms is easier. So if you take, this is my drawing for fish, yeah. This fish having all the microorganisms on the surface, yeah. And if you give a plasma treatment, you are only killing the microorganisms on the surface. These microorganisms may not be present inside the flesh. Yes, they might be present in the viscera and other one, but it will not be there inside the flesh, yeah. If they are in the flesh, that means they have some disease or internal infection. Yeah. Did I answer the question?
Yes, sir. And what about the lipids? What about the lipid oxidation and like
Again,
peroxide value of that? Because
yeah,
most of the lipids will be on the upper surface. So
no,
if it
not really, no. It's actually if lipids surface, okay, it can be on surface and all. Oxidation can be triggered. Yeah. That means there could be some free radicals, but there are certain natural mechanism of quenching those free radicals. But with the plasma alone, certain free radicals which are formed, they are for microseconds or milliseconds, which quickly goes to the stable condition. Yeah. It can promote, it can trigger oxidation, that not only for lipids but also for other nutrients. But again, use of plasma for high fat food is if you do the plasma treatment with milk, will have completely different smell. Plasma treatment of surface decontamination, that won't be that issue before lipid oxidation, and has been investigated, and even for pure oil purposes, for pure protein, oxidative products are much less compared to what we use it for thermal, and it does cause an oxidation. I am not denying that it won't cause an oxidation, but level of oxidation is less compared to other technology. So, when you are evaluating any technology for any given product, first the microbial, second one is the nutrient, and each compound, one technology cannot work for all the systems. Plasma is okay for surface decontamination, but will not be good for liquid food, will not be good for milk. Even though you would have seen a lot of studies are there for plasma for milk treatment and all, but is not going to see the commercial application. Same thing applies for UV light. UV is okay for surface treatment, but only for surface treatment because it does not penetrate in. Yeah, and plasma formation and ozone, if you are using only oxygen, anyway we will be covering the next one in the plasma. If you are using only oxygen, you get oxidative species. Yeah, whereas when you are using air, you get number of different reactive species. Yeah, I'll cover those plasma. Then if answer is not clear, you can ask that question again. Yeah, next
I
one
have
please.
another question.
Sorry, go on. Yeah, next question.
So regarding the production of duckweed in like excretes of animals, like you said, we can grow duckweed in cow dung slurry and even swine slurry, swine excretes. So what about the harvesting techniques for duckweed in that slurry, because it will be like it will not be like in wastewater, right? So it will be like it will be a sludge. So how can we harvest it?
Okay, now this is this is good question, because if you would have seen, have you seen how the slurries are handled at the dairy farms? Have you been to dairy farm? Have you been to swine farms and all?
I have been to dairy farms.
Have you seen a dairy farm? Okay. Dairy cows, one is free grazing, which is on the grass side. Yeah. Other one is stored in a particular stuff. Yeah. What happens? There all the slurry, that is poo and pee. Yeah. Both are there, and it is cleaned over regular time. Right. So that means the suspension, what is there, having a both solid and liquid in it, plus chlorine water and other things. That's the reality. Yeah. So what happens first is the solid portion is separated. At the moment what farmers they do, they put it at one place and leave it, and it fertilizes and just do creates a fertilizer. Yeah. But certain part of the world where this is intensive, yeah, like in Ireland and certain European countries, where intensive animal farming is done. So there is slurry spreading is one way, but it releases a lot of methane and ammonia and other things into environment. So when that slurry is there, water is separated out, liquid portion separated out, and the solid, which is used for fertilizer purpose. When you have that particular water, and here we are also talking about the wash water. One place is slurry is separated and the wash water is the other one. Whereas in here, whatever dairy farm I have seen, slurry, wash water, everything is put together. But whereas mechanized system, these things are separated, and that particular water contains lot of nitrogen and phosphorus. If you dump it directly to the sea, it will cause eutrophication. The purpose was those water to use it into a system to grow duckweed, so that that inorganic nitrogen, inorganic phosphorus can be converted into the protein. So if that water is used in terrestrial crop, perfectly fine, you are putting in a fertilizer. But we do not have a land A, we do not have enough of a space to grow more and more crops, and whereas duckweed gives an alternative to produce landless cultivation. If you have seen that particular figure, oat, barley, sorghum and soybean, and what is the land requirement for duckweed. So you are solving two problems at the same time, and with these duckweed they grow at much faster rate, like in 24 to 48 hours biomass is doubled, and that means you are also creating an alternative source of protein which can be fed back to animal as a source of protein.
Okay, so anyhow that solid waste will be a waste.
Yeah. So there will be, and then won't be waste, will go for biofertilizer or so.
So anyway, it is going to create methane and other things too.
That will be there definitely. That's why it's actually balanced. You can't take everything out of it. Yeah.
Okay.
The bigger problem for methane emission in animals is because of mulching. It's not because of feces and or slurry. It's slurry is an issue, but at the same methane emission coming in from mouth, yeah, A, and that is again the natural animal. It will produce a methane and CO2. Another challenge here was why duckweed was introduced, because the animal feed was coming in from soybean, and soybean cultivation is increasing at the expenses of forests. Yeah. So, in order to eliminate the dependencies on the soybean, the new biomass, new alternative proteins are coming into business. Yeah. So, these are the number of options. How do we get other protein? Is there any other source of plant protein? We need protein for animals, we need protein for humans. So, how do we make a proper balance? So, that is all the purpose. So, we cannot, if either we focus exclusively on environment, forget about economy and forget about humans, or else we have to make a balance between economy, ecology and environment.
Thank you, sir.
Yeah.
Sir, we are talking about duckweed as alternate protein source. How is the bioavailability of the protein content in the duckweed?
That's good. That's again duckweed protein is there, A, and second thing related to bioactivity is similar will be as far as other proteins is concerned, and it's been lot of investigations are ongoing at the moment to estimate how bioactive it is to make it into the feed formulation, yeah, or also for human food. There are certain studies are there, and if bioactivity largely depends how the protein is processed and how we are extracting the protein. So, their bioactivity is very similar to other plants, and then again different interventions are there to improve the bioactivity, digestibility or bioavailability or bioactivity. The three things there. What are the three terms related to that? One is bioavailable, yeah, bioaccessibility and bioactivity. Yeah, the three words. Whether the protein whichever is present, it is available, that means protein, that means it is digested into amino acid, yeah, and it is accessible at the required system. And third one is whatever is absorbed in the form of peptides or in a form of amino acid, they do have the same bioactivity. Yeah. So, the works are ongoing and there are different techniques to improve those properties, very similar to wheat protein. How do we improve the digestibility of wheat protein, or how do we improve the bioavailability of wheat protein? So, this questions it can be answered that way. It will be difficult for me again to say what will be the bioavailability, that will be a wrong answer. Yeah.
Okay, sir. Sir, if I am planning to cultivate duckweed in a smaller scale in my house for my household wastewater, how much will be the yield? What are the requirements? Whether it is cost efficient, whether the cost I am spending in that and the amount I am getting from that will get whether will I get any profit from that?
Okay. Now, what I was showing here, to grow duckweed is easier. Yeah. How you can grow it. The question what you are asking is as if I have done the business case, I know all the economic model. Yeah. Cost of growing of duckweed, what will be the yield, that is all has to be optimized based on the biomass what you are going to produce and how much you are going to get. Yeah. So, for that you need to do, for that you need to do all techno-economic assessment of the full process and then only make a business case. Yeah. So, what you see the duckweed production commercially is being carried out, as you increase the level of production, the cost automatically reduces. Yeah. So, that can be grown at home, and you have to see how what type of wastewater you are growing, what temperature condition you are putting in, all those factors will come into play. Yeah.
Thank you, sir.
Sir, instead of extracting chitosan from lobster, shrimp shells separately, what about if we combine all these into a single biomass and we do extraction, will the yield be same?
Well, definitely you can do all the things together, and then in that case what you have to do, the best strategy will be to optimize the individual one, yeah, and to see what is the best condition which can operate on all three. Yeah. For example, if you are those who are extracting plant protein, yeah, we know cereal protein is good, right? No, it's not good, lacks one amino acid. Yeah. And legumes, they lack one amino acid. Yeah. If you mix both the cereals and do the co-extraction, the protein what you will get is a perfect amino acid score, right? So same way when you have 3 or 4 different biomasses, if your target is for chitin extraction, shrimps will be easy to solubilize, yeah, compared to crabs, yeah. So it may be economical when you are doing with the crab, so you take those conditions of crab and then shrimps can be added in, yeah. So or lobster. So depending upon the process, it is definitely it is possible. Again, yield depends upon how much it is extracted from different biomasses, yeah. But if you mix crab and shrimp, shrimp dissolves faster compared to crabs, yeah. If you take a shrimp shells, then you are not able to demineralize this completely, then it will be compromising the purity of your sample. So you get the best condition for shrimp, yeah, get the best condition for crab, whichever one is higher, use those ones for each step, yeah. And this is the reality actually to have a mixed waste, because when you talk about fruit and vegetable waste, is not a carrot is one side, cauliflower is the other one. When we get from the processing system for fruit and vegetable industry, we get everything as a mixed waste. So if mixed waste strategy is there, that's much better.
So what about if we grind it completely and then we do for extraction?
No, it's still like demineralization will be, when we are grinding it, then efficiency is improved, but still when we are grinding it, we are not separating the calcium network from the protein and chitin. We are talking about biomolecule, and there are smaller molecule which cannot be seen with the eyes. When you see a powder, that does not mean it's a chitin, you know, like we are talking about the molecules. And then again it has to be, calcium has to be removed, demineralization has to be carried out. And in these cases you have to grind it anyway. Then also even if you grind it, demineralization time for shrimp will be shorter compared to crabs, because the network is like that, yeah. It's something like cement is also a powder, wheat flour is also a powder. They both are the same particle size, but you cannot break cement as faster as wheat flour. I know that's a wrong combination, but take it in that sense, yeah. Particle size reduces the particle size to improve the surface area, but when you are dealing with the molecular level, it becomes different, yeah.
Good morning, sir.
Good morning,
so
sir.
In Pyro Tower project, duckweed is used in a feed for dairy farm. Likewise, can we use microalgae for the feed for dairy farm?
Yeah.
Micro and macroalgae.
Both microalgae and macroalgae both are used for, that was one of the option, yeah.
In your circular economy, can we use the micro and macroalgae?
You can use microalgae also. We have also macroalgae for land-based cultivation and duckweed, and there are other aquatic plants. There are more than 15 to 20 aquatic plants which are emerging again to use it as an investigation. You don't have to just follow the duckweed. If you go near the lake and to see what plant is growing faster, harvest it from the local environment, like what think 1000 years ago when wheat plant was domesticated. No, we domesticated animals, we domesticated plants. Same way, go in local environment, see what plants are growing, take it out, see its composition, do the profiling and everything. So, why to go for duckweed, why to go for microalgae, we will grow this plant, yeah. That's the only way we found lot of crops, yeah. Then you can come up with a novel crop. Don't just get stuck with the duckweed, yeah. Duckweed is one, water hyacinth is there, you know, you see all those lakes with the big green plants. So any of those plants can be grown, and you might be able to have a much better species compared to microalgae or macroalgae or for duckweed, yeah.
Good morning, sir. Actually,
morning.
my doubt is about the polyphenol contents in duckweed. Like a 2H SWOT polyphenol also has both positive and negative effects. Like at a higher concentration, it will give a anti-cancer and anti-inflammatory effect. But even at the higher concentration, it will give more anti-nutrition effects and also impart a bitter taste to the food. So duckweed bitterness. So it will impart it will impart bitterness to the foods. So duckweed is usually preferred as a cattle feed. How can we construct a design of experiment to optimize the balance profile?
Now, one thing is, I don't know which paper did you see where you have seen there are a lot of, that is again the number of different duckweed species are there, right. So, Lamna minor, Lamna gibba, Wolfia and different species of duckweed are there, yeah. So, the growth of that duckweed depending upon if you are planning to use that duckweed only for animal feed, then you have to select a certain species. Yeah, and it's very similar way to answer if you're growing the wheat crop, whether you are using that wheat crop to make chapatis or to make a bread wheat flour. That is Triticum aestivum, Triticum monococcum, Triticum dorum. Same way the rice. Yeah, rice, if you are using basmati rice to make an idli, what will happen, you know it. Yeah, same way if you want to get to make a vegetable biryani, and if you use parboiled rice or the small pony rice, what will happen. Yeah, so that is again the crop what is grown or species what is grown, that largely depends again what is your intended application. Coming back to a phytochemical, it does contain phytochemicals. The name itself is phytochemical, the chemical present in plants, correct. So those, when we say phytochemical, even certain compounds are toxin, they are also any chemical coming from plant is known as phytochemical. There could be a food phytochemicals which can have a food application, or phytochemical which will have non-food application and work as a chemical. Yeah, because a lot of drugs and medicines are also obtained from plants as an antimicrobial, antibacterial agent. And then if you separate it, you purify it, and then toxicity depends upon that. Caffeine is present in a coffee, we all drink a coffee, but if you concentrate the caffeine and it becomes a toxic one, or if you put it in a tablet along with ibuprofen, it acts as a painkiller. Yeah, so that is all depends upon the concentration and the species you intend to grow.
Thank you, sir.
No, madam, she is waiting for her time for a long time. Yeah, you go on. Yeah.
Yes, sir. My doubt is on reverse osmosis of water, sir. Even though it filters sediments like some calcium carbonate, sodium carbonate, there is a wastage of water, sir. Always there is a wastage of water. Why it is so? Is there any alternative techniques to utilize that wastewater other than utilizing it from in domestic level?
Now you see, when we are using reverse osmosis, is where is the wastage of water? You just remove the minerals out of it. Yeah.
Yes, sir. There is always an leftover water there while processing, while filtration.
Yeah, that's the reason I was saying, like, we know, if we get a complete zero, means zero waste, one is perfect. Yeah, but there will be always a side stream and the wastewater coming out of it. Yeah, but in reverse osmosis, when you are using it, the only leftover part is your minerals and some concentrated water, because mineral cannot be separated just like metals. Yeah, and that particular water is having a rich source of mineral, calcium, phosphorus, and other one. And if we do know the composition of that one, and then try to see where else we need to use it. I'll give you an example. If suppose I'll bring you back to the shells, when we do cells dissolution, what we get? CO2. Yeah, and then if you want to make a circular model, this CO2 can be used for microalgae cultivation. CO2 is utilized. A. This is best case scenario. In this one, what else we get? CaCl2. Yeah, and the calcium chloride is used for as a filtering aid. Yeah, other one usage of calcium chloride is to make your drinkable water. Yeah, so it's again, when we are trying to utilize it, it's easy to make full biorefinery on the system to say it's zero waste. So at least if we are achieving trying to achieve zero waste, at least we are making best use of whatever available resources are. And I'm pretty sure the water whatever comes out of RO, they have lot of minerals and the salt, and certain minerals can be easily recovered, like salt, you know, like natural sea water, we get a salt. Yeah, and then in that particular salt we are using only evaporation to leave the moisture up. Yeah, and then if you prepare a closed system, then you can get a drinking water. The one way is the desalination, to remove the salt and the sand from the water. Other one is to, normal sunlight is there, we create evaporation, and that leads to again water cycle and rain and other thing. But if you have to get the drinking water from ocean, and then you just attach a distillation system, you get your right water. If you'll see some forest, certain trees, you make certain mark and you collect tree sap, like rubber plant, another one, that some plants they you get large amount of drinkable water. So that will be there, and you can definitely make use of it, even by having higher amount of mineral, if you see if this water is good enough to grow other plants. Even for duckweed you need certain nutrients. If you grow on RO water, it won't grow. If you make a batter system I'll give completely different example. If you make a batter, you know, for dosa or idli, when you make a batter, if you try RO water, tap water and deionized water, which one will give the best batter? Tap water, yeah, because you need to have those minerals. Have you ever tried that? If you try deionized water, go home, you get the deionized water from the lab and try to make a batter, it won't form a batter, because you need to have a natural microflora and you need to have certain nutrients like that, yeah. So simple answer is to see what is the composition of that and then to see where all it can be used, yeah. That's the point where you have to use a literature. Composition of water is mineral, phosphorus and all, then you have to see where you need a calcium or phosphorus, and if you will see globally there is a shortage of phosphorus. Globally we will see the mineral wise we are in short of phosphorus, but at the same time we have a phosphorus from biological sources. So that way it can be established.
Thank you, sir.
Good morning, sir. Sir, my question is the chitosan produced from the crab cells and prawn cells will satisfy the all good packaging ability as compared to commercial packaging materials?
Yeah, so I'll have the packaging one for chitosan. I'll have in the next lecture we're going to go through a biopolymer for making packaging, yeah. So chitosan is good for biopolymer, yeah. Is any of the molecular compound which is having larger molecular weight can be used for making biopolymer, and chitosan we are using commercially also to make a alternative packaging material. That was your question, right?
Yes.
Yeah. So and you'll see you will have more questions when you go through the biopolymer application.
Okay, thank you, sir.
Good morning, sir. Sir, my question is, sir, you discuss more pre-treatments like ultrasounds or what about the osmotic dehydration pre-treatment, sir? Because I do my PG research in osmotic dehydration on paneer. It helps to retain texture, retain color and all other physical properties. So what about the osmotic dehydration as a pre-treatment for the seaweeds and also, sir?
Osmotic dehydration by using salt, yeah?
Yes, sir. Yes, sir.
Salt or a sugar is one of the way of pre-treating. The biggest problem even in a seaweed one is when you already having a lot of salt. So 3.3 is the soil salinity for seaweed. So we have to exceed more than 7 to 8% of salt in a seaweed to preserve it, yeah? That's not a problem to use it, but then the ingredient what we are going to get, then removal of that salt needs lot of water, yeah? So there is salting or silage is another way, is one of the way to preserve that particular biomass. But another problem is when we are getting the ingredient, the same thing is happening with NaOH and hydrochloric acid. We get salt, NaCl, because of NaOH plus HCl, yeah? So what we get is NaCl, and then the sodium content of the alternative protein increases, and the sodium itself is considered as a negative aspect of any of the product, yeah? So that is all those limitations. The selection, yes, osmotic dehydration definitely can be done, even with the sugars it can be done, but the problem is again how do we get rid of it, yeah? If you want to just preserve the small amount of seaweed with the salt, 10%, 15% or whatever, or even with the sugar like what you do pickling, yeah, and then you can preserve it, and then if you are doing it for your direct product application, perfectly fine. It's not, that's why the big diagram was saying conventional and the new ways of doing it. It is done anyway, salt preservation.
Sir, is any possible to produce like in fish salted osmotic dehydrated salted fish we consume, as the seaweed is produced from that way, like osmotic dehydrated salted seaweed is consumable for a human or any
yes, it is definitely, yeah, because some of the companies even are using directly seaweed as a pickling purposes, yeah, as a seaweed pickles. You add like what you make a lemon pickle, carrot pickle, same way seaweed pickles are also there, which gives not only now the shift is more towards fermented food, you know, kimchi and all those things, and the same thing can be prepared from seaweed. Yes, it's and then again when you ferment it, then you are getting a lot of other benefits of using different nutrients, beneficial microorganisms and everything there. Yes, it is there, and moreover some of the crackers made out of seaweed contains large amount of salt. If you use, in your case, using salt with the seaweed and then you macerate it, and then if you make a sheet, you dry it just like a papadum, and there's some of those rice seaweed crackers are made due to the presence of alginate. It forms a layer which will have a crispiness. That is there, yeah.
Sir, if I produce seaweed, like salted seaweed from the drying, after drying process, what about the rehydration ratio of that end product, sir?
Rehydration ratio. Now that is rehydration ratio will have to investigate. I will not have a clear answer how much it will be rehydrating, because rehydration ratio will be in a tone of 60 to 70%. We did do the drying of seaweed by using different techniques. For freeze drying, you get almost everything. Yeah, nearly about 100% to 90 to 100% of rehydration ratio. Go for high temperature hot air drying, maybe 30 to 40%. So rehydration ratio has to work out how much it is. But when you are using a salt to take the water out, then you have to remove the salt to get the water in. It depends upon how complex the structure it becomes. Yeah.
Sir, another one question from my own research, sir. Actually, I did osmotic dehydrated paneer, sir, and I used the paneer to produce paneer gravy, retard paneer gravy. After storage study, we have studied mineral content is increase the oxidation process of lipids, increase the lipid oxidation, but my product which have solute concentration, it reduce the oxidation compared to the control one, that fresh one. How it possible? I don't know.
So osmotically dehydrated, because you are, if you see, it will be very difficult to establish now why lipid oxidation is reduced, and this lipid oxidation is measured by using T-bars.
TBA, sir. Thiobarbituric acids, free fatty acid peroxide value.
TBA or T-bars is there, and other one is hexanal formation by using HPLC. There is also some limitations are also there with the type of method used for measuring the lipid oxidation. Yeah. Because T-bars is thiobaritic acid assay. This is the most common method used for lipid oxidation, that is for global lipid oxidation. But most important one comes in from peroxide value and also by using GCMS, hexanal, short chain fatty acids. So what could be the possible reason when you are using the salting one is that you are draining out the water. So you are avoiding hydrolytic rensidity. Yeah. See, when we talk about lipid oxidation, is not only because of air. Lipid oxidation is also because of water or for oxygen. Yeah. So hydrolytic rensidity might be avoided when you are using your osmotic one. That means you are draining out the free water, which might be causing lipid oxidation.
Sir, but at the storage period, my paneer soaked in the gravy, sir. Gravy contains also water.
No, correct, but the one which causes the lipid oxidation in the protein matrix, as I said, like lipids are not present in isolation. Protein is mixed with the lipid. That's the way the molecule structure comes in. If lipid is separate, protein is separate, extraction technology becomes easier. Just put a hand, put the protein in the side and take the lipid out. Yeah. So it's a network of a molecule. So that could be one of the possible reasons. Then if you go, when you are using lipid oxidation, try to measure your hydrolytic rensidity and oxidative rensidity. Then you will be able to quantify which type of rensidity and oxidative stability has been compromised with. Yeah. So that number of different methods are there to measure free fatty acid which are resulting from hydrolytic rensidity or from oxidative rensidity. Then you will be able to quantify easily why your osmotic one is there. That's different story. Afterwards you add a water, because oxidation is within the protein network. Yeah.
Okay. Thank you, sir.
Sir, my question is, which type of a strain we can use in laboratory?
Strain of?
Sir, related to algae, cholera and other species.
Okay, now any of the strain depending upon again on the applications, and moreover whatever is well studied. Suppose you want to study number of different types of single strain and to see how it works on a biomass, any experiment carried out, you have to only vary one variable at a time. Either you vary the different types of strain performance, yeah, or different species performance, or test the same strain on different type of biomass conditions. So again, that strain one depends upon what will be end product. If suppose there is a genetically modified strains are also there, but then the product comes in will be genetically modified, then you have to fight through other regulations. So and even depending upon what performs best on your on the biomass.
Good morning, sir. Sir, my question regarding to the algae culture. So there is different methods like heterotrophic, mixotrophic, autotrophic. So if we cultivate algae in our lab, so which one will the best?
Now this is again the question, because if you want to make a complicated one, it's again the mixed culture, single culture, or with the light or different oxidation. That depends again, you know, like how, what is the strain you are picking, as your friend asked the question, what strain you are picking and what are their requirement and what is the best condition for that particular strain to grow. Yeah, that will give you the best selection of the method. Yeah, so most of the work for each and every strain is already been carried out. If suppose you want to have, you don't want to purge external oxygen, yeah, then you have to pick certain type of strain. So objective is to grow a specific type of microalgae, or objective is to valorize the wastewater. Yeah, for valorization of wastewater, that means you want to have a minimum input. You don't want to put a specific UV LED light system of 35 nanometer or so to make a complicated system to valorize the wastewater. Yeah, so to go with the best system which and pick the strain and the condition which is much more economical and convert the waste nutrients into useful nutrients.
Sir, there are lots of media for algae culture, just like VG11, F12, BVM. Sir, which one is the best for the high biomass yield?
Now, it's actually here the problem is you have to use medium as a wastewater, right? You know, because there are best media is there for microorganisms to grow. Yeah, for instance, even forget for the moment microalgae, take lactobacillus. The best media is there to have a specific type of agar, nutrients and all. But we use that one into milk to grow, make yogurt. Yeah, but milk will not have all those nutrients which are required for a specific media to grow. Yeah, same thing, microalgae will be best performing on a specific type of media. But here your objective of your study is to use wastewater as a media for microalgae to grow. So performance won't be as good as what is there on standard media. Yeah, it's something like, you know, best standard condition we operate the best 25 degrees Celsius, but conditions outside may not be 25, maybe 10 degree or maybe 45 degree. So what will be still will perform, not to the level what we will be doing at 25. So here in this case your objective is to valorize the wastewater. Yeah. So that's why you have to see the media whichever matches that particular wastewater. Otherwise, you will be adding more and more nutrient to the wastewater, and then purpose of your study is defeated.
Sir, last question. Sir, we know about the just like the positive impact of algae, different application. So there will be a negative impact also. Please explain that.
Yeah, negative impact is when you are using wastewater along with your microalgae to grow. Suppose, and then you want to use that microalgae for food purpose. A, regulation hurdles. B, if your wastewater contains a lot of heavy metal, because wastewater over here is not the same as agriculture wastewater or industrial wastewater, and suppose mercury and everything is there, then again you are loading all the toxic element into microalgae and you may not be able to use that biomass for other purposes. Second hurdle. Third one, again for downstream processing, that means how you remove microalgal cells from the water. Yeah, yes. Even if you remove it and if you dry it, whether it is worth to grow microalgae or to just go with the sludge system. Yeah, so far what we were talking all about blue circular economy was only the food waste side of it, or for the process water. I classified in between whether we are talking about the process water or wastewater. Yeah, this wastewater is mixture of sanitizer and everything used in in food industry premises. So this goes for, in the case of fish, we had a stick water, yeah, whereas processed water, which is used for washing a fillet, production of protein. If this goes for microalgae or duckweed production, then you are okay to use it for food purpose. When you are doing with the wastewater, then you have the biomass which is produced, then you can only talk as a phytoremediation, yeah. Phytoremediation, that means using microalgae to clear the wastewater rather than creating the sludge. Second one is to reduce the BOD and CO2, BOD and COD, so that your impact on wastewater treatment is reduced, yeah.
Sir, so we cultivate algae with the help of wastewater. So we can't use as a feed for any animal.
That has to go through the regulation.
Okay.
Any food, anything grown and intended for food or feed purpose, that has to go through a specific regulation, yeah. But that's a third stage question. First stage itself is to see whether microalgae can be grown or not, yeah. It is something like this type of question, you know, about regulatory approvals. Another one comes in to see how we can improve the process and what are the conditions to grow. The regulation says for any feed you need to have A, B and C and D. Even heavy metal specifications are written. It cannot be more than 50 ppm, yeah. So if it is more than that, it cannot be used. Not only for that microalgae, that is true for almost any biomasses. Even if you grow a peanut, if it's having certain level of mycotoxin, it can't be sold, yeah. So that is all a number of stages there. So that's why I think that will be coming in today about the TRL. So before we start doing the experiment, that is proving the concept, what your intention here is to prove the concept, yeah. And then looking straightway mission and deployment, that is quite different. So technology upgrade happens in a sequential manner, yeah. So your task first one is to see whether wastewater can be valorized by using microalgae, A. Second one, what type of biomass are there, how much BOD and COD can be reduced. Hypothesis is, yes, microalgae can be used to valorize wastewater and it can reduce BOD and COD. Now you prove that hypothesis or you reject that hypothesis, yeah. If we know all the answer, then there is no point in doing it, just straight away we execute it.
Okay.
Yeah.
Thank you.
See, the problem is when we start doing any experiment, straight away we think, oh wow, we'll make this, you know. There was a story, two couples were fighting and the third person said, why you are fighting? So we were discussing, we are going to buy a cow and who will make cheese out of it or will make sweet out of the milk. And then based on that argument, they both were fighting. The neighbor, he started beating both of them. He said, why are you beating us? Because your cow will be there, then your cow will eat my grass. But we don't have a cow. I say, yeah, you don't have a cow. Then you are fighting for whether they'll make sweet or a cheese. Yeah. So that's before putting our first leg, we should not lift our second leg. Yeah. So that's I know that's good to think to have a long-term vision. But at the same time, we have to go for step by step onto it. Yeah. Yes, sir. You're the last one. If no question, that's okay. Oh, it's almost 11.
Sir, shall I know about Brijesh Tiwari, sir, biography?
He's whosoever was giving the introduction, you missed the first day, right? So he'll be able to give it to you. Yeah.
Biography in the sense I want to know about why, what, how. Why Tiwari sir is passionate in the field of science. What are the studies he undergone? The process began him to come the process began him before you come and sit the studies you undergone, sir. How? What are the things we can implement in our career life which is helpful for me and my colleague?
It's a difficult question to say. Yeah. So it will take a lot of time to explain if I say biography, because I'm in early 40s, so 40 years lifetime will be long to explain. Now coming back to your question about the last one about the career and all, you should do what you like to do. Yeah, you know, you should study what you like to study, not to study what other people are studying. And during those questions I came, yeah.
In the first day, I hear one word from you, sir. Dressing like someone, copying someone. I want to dress like. I want to find similarities which can help my. I know about. We want to study. We want to pursue what we like, what we passionate. But I want to know about you to find similarity which can helpful for me and my colleague.
Similarity.
Why you passionate in the field of science?
Well, see, basically, if you it's not only the science. If any area what you are working on, if you are interested, if you make work as your hobby, then you will never be tired. Yeah. If you start thinking that you have to go 9 o'clock, Monday 9 o'clock, how many people they have a problem? Yeah. Only few people they think Monday 9 o'clock is good. Today I'll do X, Y, and Z. Yeah. So it's all when you take science rather than your work. Yeah. Or you take it as your hobby or a passion. Yeah. That makes lot of difference. Yeah.
But what are the studies you have learned?
Okay. I did my same 10th and 12th I studied in basically like what we all study, science. Yeah. And then Bachelor of Agriculture and Animal Husbandry from UP Pannagar. Then Masters in Food Technology. Yeah. And I'll be very honest, till this point I just studied whatever I got on my way. Yeah. There was no intention that I want to do BSc Agriculture or want to do Masters. Yeah. After that I was working in industry for 6 months. I got bored for a routine job, went for currently ISCPT.
Slow and steady.
Yeah.
Like you teaching in the class.
Okay. So what we'll do is I'll talk to you in person and I can explain you everything. Yeah. Okay. Because now we have to take a short break and we'll come back. Yeah.
So no, nothing problem, sir. You explain the fast manner.
No. We'll come back again, and after the break we can talk again. Okay. Yeah. This one, screen. Is it visible? Okay, reduce the light. Is it visible? Oh, turning on. Okay. Okay, so we'll start. Okay. So we'll continue with novel ingredients and technologies for food application, and the next one is going to be feed application. So crux of the stuff is again related to ingredients and the product. So we'll start with the technology what you also have the demonstration of. So what is cold plasma technology? Yeah. So cold word itself is quite ambiguous. Yeah. Cold does not mean it's very cold. That means it's not increasing the temperature of the product by a certain degrees, maximum 2 to 3 degrees Celsius. But there are other plasma systems which are also considered as a hot plasma system, where the temperature of the product can increase significantly. So if you see the state of the matter, we always have a solid, liquid and a gas. Yeah. Certain products they convert directly solid to gas. Example, camphor. Yeah. Water, ice converts to water, water to water vapor. Yeah. And if we break the gas, it becomes OH and H plus. So these are known as ions. Yeah. So this is what state is known as plasma. Yeah. So these are called also free radicals. Yeah. So they are free so that they can oxidize it, because everybody has to come at a stable position. So plasma, an energetic ionized gas which is widely used for industrial material processing. Now in food also we started having different system. So this is one of the way how we can reduce the microorganisms. So any of the gas which is broken down into ions makes it a plasma. Yeah. So how we can break the gases? That's a challenge. One, you take any neutral gas, whether it's air, CO2, oxygen, hydrogen, helium, nitrogen, any of those gases, we give heat treatment. If you heat it, heat the gas, it can be broken down. It's just not normal boiling the gas, it will convert. Electric, that is voltage, magnetic field, radio or microwave frequencies. Any of those techniques can break the gases into ions. Yeah. So then it becomes ions and then you get your plasma. So these can be known as reactive oxygen species or reactive nitrogen species. Yeah. Now here, if I have only nitrogen as a gas, what reactive species will be formed? Nothing. You need to have an oxygen to form nitrates or nitrites. Yeah. Same way, if you have only CO2, it will be difficult to form reactive species. But if you use only oxygen, what will form is ozone or ozone related compounds. Yeah. So that is the difference. What type of reactive species are formed, that largely depends upon the type of gas used. Plus certain reactive species are also formed depending upon what biomass you are treating. If biomass contains moisture, moisture provides H2O, that is vapor. Vapor along with nitrogen can also form nitrate, nitrites, hydroxyl radicals. So chemistry is quite complicated. Yeah. It's not that easy that you put in a plasma and see how it is operating. Yeah. Now lot of studies, if you'll see, it says 10 kVA plasma, this is the inactivation, 20 kVA, this is the inactivation. Technically it is correct, but scientifically it is not correct. 2 gram poison can kill me, 3 gram poison can also kill me, right? So there the concentration of poison on death of any individual is not proportional. Yeah. The same way the voltage becomes meaningless. Voltage only creates plasma species, and what is the fate of that species depends upon the gas. Yeah. As well as on the surface what you are treating. That will dictate how it works. Making a plasma system is not that complicated. You just need to have a high voltage transformer, that is step up transformer, where the voltage is increased, current goes down. Yeah. If you are able to create 10 kilovolt That means if you will see lot of electric distribution system, the big transformers, it's written 5 kV. Yeah. So from 5 kV we are getting 220 kV, sorry 220 volt. Yeah. Now here we are creating 220 volt and to get 10 kV. So you should be getting a Nobel Prize for that. Yeah. But it is not the voltage which creates or gives electric shock, it is the current which gives. Yeah. If you will see 5 amperes is the normal connection, but once you go step up transformer to increase the voltage, the current becomes low. Yeah. We are talking about in milliamps. So that's the difference how the plasma is created. Yeah. So in simple processing step, you have a packet with the gas or air in it, you put high voltage discharge, whatever gases are present here, they convert into plasma species and then they oxidize the microbial organisms in terms of oxidation of their surfaces, peptidoglycan layer and all those stuff can be oxidized, and that way it makes product more stable. It is as simple as that. Yeah. Now when it comes to application, so now you are clear what a plasma is. Yeah. It can be hot plasma, thermal plasma, microwave plasma or RF plasma, all these different types of plasma created based on the source. Yeah. Inactivation mechanism is oxidation. What causes oxidation is free radicals. Yeah. So free radicals are there, and in order to quench free radical, what we use called antioxidant, your phytochemicals, another one, right? But those two things cannot be related. So that means if you have a phenolic solution and you give a plasma treatment, it will oxidize those phenolics. Yeah. So that's the way it operates. So this is all free radical which causes application, which causes inactivation of microorganism and also responsible for extraction purposes. How? We'll find it out now. Application, pre-harvest, post-harvest, even processing. Yeah. If we have free radicals and if we have a wastewater which is having higher amount of BOD and COD, the moment we give a plasma treatment, it will reduce its BOD and COD because oxidation of organic matter. Yeah. So that is also one way of treating the water. UV treatment of water you have studied. Yeah. UV also forms certain amount of ozone and free radicals. Yeah. Have you seen when there is immediately after a rainbow or immediately after a rain, there is some nice smell is there, a nice environment is there. That is also because small amount of ozone is produced. Yeah. Ozone in small amount is good, in large quantity is bad. Same story is true for everything. Neither excessive rain nor drought is good. Yeah. So in same way, at optimized concentration we can have certain inactivation or activation. So when it comes to application, I'll quickly go through all these ones, right? Microorganisms cause for inactivation. Yeah. Fresh produce, surface cleaning, surface decontamination. Grains, again microbial decontamination of molds and fungi, and if you use a plasma along with the drying, so you can get rid of those fungal infections so that mycotoxins are not produced. Even though if mycotoxins are produced, plasma can degrade those mycotoxins to a certain extent. Fish, same thing, plasma activated water, if fish is frozen, it extends its shelf life because the free radicals are present, and free radical life also depends upon the temperature, at what temperature it is there. Half life of ozone at 20 degrees Celsius is almost 20 minutes. That means ozone 50% goes back to the original state in within 20 minutes. Ozone is O3, that means O, right? Automatically it needs something to stabilize, either with 2 molecules of ozone will make 3 molecules of oxygen. Yeah. And then or else you have hydrogen or other thing, if it does not get stable state, it will oxidize certain matter. Yeah. Principle is very straightforward. Dry food, again we can fumigate with ozone. If you will see the other day when we are talking about the contact and non-contact type, this application will be non-contact type. Meat, again application is there, but here the question of your about lipid oxidation, if meat surface is having meat slices, they have a fat. it will have more impact on lipid oxidation compared to whole of the chicken carcass. Same thing, whole of the fish will not have much of lipid oxidation compared to fish fillet. So, these are some of those limitations. Where to use, when to use becomes an important aspect. Same thing for pesticides. Some of those plasma, since they have free radicals, and majority of the pesticides also they work on free radicals. Sodium hypochlorite, yeah, that is nascent oxygen is the one which causes most of the microbial inactivation, yeah. Same way, there are different mechanisms are there. We can replace the pesticides by using plasma. Give you simple example, we all need oxygen to survive, yeah. If I oxidize that oxygen air with the free radicals, that is what all the pollution story is, our lungs will get dysfunctional. That means, if you have insects and if you are using a plasma, insects will breathe plasma species, it will oxidize their organs and they will never develop resistance against it, because you cannot develop a resistance or allergy against oxygen, yeah. Can we? If you get allergy against oxygen, then that is the end, yeah. So, when it says pesticide, that is your, this is a broader term used for what? Insecticide to kill insects, yeah. Acaricide to kill aphids and other organisms, bactericide to kill bacteria, fungicide to kill fungi, yeah. So, together it is pesticide, yeah, that is pest. And food industry operations for replacement of chlorine for washing purposes, reducing a BOD and COD or different unit operations. So, there are lot of different applications are there, yeah. Now, this is much more description on plasma. Microbial decontamination, insect control, I just gave an example, and improve the flour properties. If you know about the starch, you have know about native starch, which is native presence. Other one is your oxidized starch, yeah, having different applications. So, oxidation starch is normally done by sulfuric acid, hydrochloric acid, different chemical treatment. So, you can use a plasma to modify the starch properties either for non-food application for reducing its viscosity, yeah. Since it oxidizes, so it also reduces its molecular weight. So, application depends upon if you have a clarity of its mode of action and mechanism, then you can find its application, yeah. Otherwise, you will say, oh, somebody used in starch, somebody used in flour. So, like that applications are developed. If you see the wheat milling process, your wheat grain, you wash it with the water, you grind it, you make a flour, yeah. Commercially, same wheat grain is washed with the chlorine water to get rid of microorganism, made into the flour, and then different products are made. So, instead of using chlorine, you can adopt plasma to wash it, yeah. At the same time, if you know the way grains are stored in big silo and the grains are aerated. Why they are aerated? Because if there are certain microorganisms left over, they produces CO2. CO2 increases the temperature and humidity and grains start spoiling and then insect infestation happens. But if you are flowing through the plasma air, so insect growth will not be there and it will help preserving the grains for longer time, yeah. You know about FCI go down, Food Corporation of India go down, they have phosphine and different gases, yeah, is circulated through the grains to avoid all those contaminations. So, that can be replaced by using plasma. And what are the residues of plasma? Nothing, just stable gases, yeah. Again, dough rheology, spices and herbs is an important application here, because majority herbs and spices, they are processed at very low temperature. You do not use high amount of heat. If you use a high amount of heat, they will not have a flavor or their bioactive content is reduced. So, if you are giving a plasma treatment along with the low temperature drying, you are able to get rid of microorganisms along with it, yeah. Fruit and vegetable, again surface decontamination, food preservation, essential oil extraction, that is again this is some bit of extraction application in terms of disrupting the cells through oxidative mechanism and opening up the structure, and you can use it for extraction purposes. Other one is the soil one, yeah. So, if the soil is having contamination, you haven't seen soil remediation programs. There are a lot of places when especially the area where all the sludges are kept, that soil is reclaimed, because the way we are contaminating the soil, in next 20 to 30 years there won't be proper soil to grow crops also. So, soil remediation program is, those who have studied agronomy, 1 hectare of land, what if topsoil, that is 15 centimeter or 15.7 centimeter is the only topsoil which is responsible for cultivation. If topsoil is gone, you cannot grow a crop. Yeah, it becomes what do you call that, barren land, because the topsoil is gone. And sometimes soils are reclaimed and the topsoils are also sold in a market to regenerate the farms. Yeah, regeneration of farm or a wasteland takes centuries to do it. Yeah, so that's the one of the reason why we are moving to more towards organic cultivation. So that's antibiotic heavy metal removal. That is one way how we can remove heavy metal is suppose there is lead. Yeah, lead oxide is a form of a heavy metal that can be formed because of plasma, and then you can remove it. There was a question how we can remove heavy metals. Yeah, so that is one of the technique how we can make into a metal form. Yeah, see, one heavy metal we talk about mercury. Mercury available in thermometer, which is obviously now phased out, is not Hg, it's an element. It's mercury oxide, mercury this one. So you convert into oxides to use those heavy metal and separate it. And it is not that heavy metal is not used. Heavy metals are used in a lot of different applications. Like car engines, they used to have a lead in it. Yeah, but now it's lead is removed from the oil just because of heavy metal. So that is one way of oxidizing the minerals to come into a mineral form. Calcium, how calcium is present in natural environment? In a form of lime, C calcium hydroxide, yeah, or calcium carbonate. Calcium is not present just like that Ca2 plus, no, yeah, it is present in a form of oxides. So you have calcium chloride, what you formed, calcium carbonate or calcium hydroxide. Yeah, same way we can also get lead oxide, lead hydroxide, and these type of things can be formed also with the plasma. Now don't think that plasma is a universal solution to get rid of all the heavy metal, no. Yeah, it's not that easy. It's easy to say than to do it. Yeah, microbial contamination, again dye removal. Lot of clothing companies, you have different colors dye. Yeah, but cotton is always white. Yeah, and those dye is used, that means clothing material is dipped into the dye and that dye is discarded. Yeah, then you have to oxidize that dye, that is again a pigment. That oxidation also happens because of plasma, or same thing happens with ozone. So you might be able to find one book called Ozone in Food Processing. Yeah, ozone in food processing. And there is also a plasma book, quite a recent one. If you want, I can send you the PDF version. This book, we wrote it back in 2009 and 10. One of the editor was Rip G. Rice. Yeah, he joined in as an editorial board at the age of 90 plus. Yeah, and he was the one who was responsible for getting ozone as a GRAS status in US FDA. Yeah. So that all the application where ozone can be used has been replaced by plasma. The reason is, for ozone we need oxygen tank, and keeping oxygen tank is very hazardous. So that's why ozone production becomes a challenge. So to replace that we have plasma, which that means you can use atmospheric air. Now pharmaceutical contamination, same thing as oxidation, effluent microbial contamination, pesticides, plasma activated water, again different application. Yeah, decontamination of seed germination, plant growth or yield. Yeah, by looking at this, it looks like the plasma is a solution for every problem, but for a specific application. This is very important to remember that. Yeah, okay. So these are different applications how we can use it, plasma, for different purposes. Now lot of systems we have designed it, lot of systems we designed it in collaboration with different industry. So what type of plasma system you have seen? This one, what type of plasma is here? Jet? That is jet, something like this. Yeah, okay. So this is also used in, we developed a system during the COVID virus. to inactivate virus, as well as in hospital industry virus is plasma is used to decontaminate the buildings and environment, yeah, because lot of things, the cold air is circulated, it's never decontaminated. I'll show you in some of the slides. So this is not exactly a microwave, it's a hot plasma. This generates a plasma with a temperature of 180 degrees Celsius. So our intention is to use this plasma jet along with the spray drying, so that we can modify the properties of the powder, as well as we can inactivate while drying, because spray drying you need a hot air 160 to 200 degrees Celsius. That's one. This is basically a plasma needles, and it generates plasma. Second thing, you can't see a plasma, yeah, you can't see ions, but what you can see it in dark, because it just creates a bluish flame, yeah. Why it is slightly bluish? Because blue is the only color which diffracts the most, yeah. Now one question to you, do you have any natural blue dye on this earth? Any natural blue color? None. Where is it? Which flower or which plant gets you the natural blue bioactive? Microalgae can do it, but depending upon the pH, yeah. Natural blue color is not there, and lot of industry they are looking for alternative to blue color, yeah. You see in the blue color drinks, yeah, you have red, white, yellow, all these colors are naturally present, but blue natural color is rare, yeah. Think over it, if you find anything just let me know. Yeah, give mic to him.
What about this
indigo?
Blueberries.
Huh?
Blueberries.
No, it's not exactly, name is blueberry, something like rasgulla. This is not like gulab jamun.
And also this blue pea flower.
Yeah. No, if you'll see that one, that color is not stable at wide range of pH. The moment you even take a strawberry, you change its pH, add an alkali to strawberry juice, it turns green. Yeah. So same way that blue color is there, but that's not exactly a blue color. It's a mixture of 2 or 3 color. Yeah. The base color, you know, RGB, red, green and blue is a base color. In that one, blue color is naturally not that available. Yeah. Sky is blue. Why? Because that's the color which diffracts the most. Yeah. So that is one of the challenge of why I am saying this, because even I was not sure of that answer. When a company came up, multinational, they are looking for natural blue color which is stable at acidic pH. Yeah. There is no fruit which is a bluish color. Blueberry and blackberry, they have some violet tinges there, but not exactly the blue color what you see in a Bombay Sapphire and all those type of drink. Yeah. Or the blue what she is having, she is wearing. Yeah. Because oh, he is not here. Blue color which keeps the evils away. Yeah. One of your colleague day one he mentioned. Yeah. So this is another plasma, and this is your plasma jet, where you have high voltage needle and the gas comes in and it forms plasma jet. Configuration, this is a ground electrode, high voltage electrode, air inlet, and it creates plasma. Now this is again variac, where you can change the voltage. That means you have variac, that means whatever input voltage, same way you can get external voltage changed, and in this case that is a dielectric barrier system. That means you have a positive electrode and a ground electrode, and between there is a gas, and it just creates a plasma inside. Yeah. So you can have any product packed inside, so you don't have to wash it and do multiple operations. This is just a configuration of equipment, and these equipment can be configured the way you want to use a plasma. Yeah. And you will see some more systems which have been developed. Application, whole fish packed in a packet, and then short half-life species inactivate microorganisms, and then they go to the stable state of air. Yeah. That is the simple process for inactivation. Plasma jet, and the same jet goes into the water, makes plasma activated water or plasma containing water, or ozone gas goes into the water, contains ozone containing water, and for surface decontamination or using it in a frozen water, yeah, freeze that water to kill to inactivate microorganisms. Yeah. If you remember when I was talking about freezing, We talked about -2 to -4 degrees Celsius, yeah, -20 degrees Celsius, yeah. So, there is one concept of freezing the fish. You do not have to keep the product at -20 degrees Celsius or -80 degrees Celsius to improve the shelf life. Simple operation is you have a fish dipped in a water and freeze it only at -2 degrees Celsius. Ice will be formed on the surface of fish. There will not be any microbial inactivation. You do not have to keep it at -29 degrees Celsius, because every 1 degree Celsius reduction involves cost. Yeah. -80 you keep it only when you are talking about those compounds which are highly sensitive to temperature, or DNA, or those ones which is required for medical application or for certain specialized application. Yeah. So, that is called super chilling. Yeah. So, that is why it is the simple concept. Why if you keep product at -8 or -20, what will be the shelf life? One place you get 20 days, another place you get 22 days. Yeah. But if you are looking for bioprocessing, it can be done at -4 also. Yeah. Because anything less than 4 degrees Celsius, it ceases its microbial activity. Yeah. And then this is the way it looks like plasma, and then different electrode system, how we can use it for fish application. Here again it comes up with your creativity, how you do it. Now, what happens? The reactive species. If you have oxygen, it generates ozone species. Yeah. If your nitrogen in combination with oxygen, you get lot of nitrogen species, nitrate, nitrites, and remember nitrate, nitrites are also the form of fertilizer which are used by plant. Yeah. There was a question about the chemical fertilizer. So, we have also seen if we have nitrates or nitrites, you sprinkle onto the plant, they get their fertilizer in the form of nitrates and nitrites. Yeah. This defines how the cereal crops need a fertilizer. Pulses, they do not need a fertilizer, because they have rhizome, root nodules which convert atmospheric nitrogen into nitrates and nitrites. Yeah. So, if we same way break the air into nitrate and nitrites, that can be used as a plant fertilizer, is not it? Yeah. And then again formation and dissociation. Now, this is related to the gases, but when these gases come in interaction with the water molecule which is present in that microenvironment of the product, then you have lot of other hydroxyl radicals. Yeah. And when you use a plasma activated water, the pH of the water drastically reduces. Yeah. It becomes acidic. Why it becomes acidic? Because there is a water, nitrate, and it forms sort of nitric acid or a carbonic acid and all. So, pH of the plasma activated water is lower. Yeah. I will throw another thing, when you are adding hydrochloric acid for dissolution of shells. So, there could be an option where plasma can also be used to dissolve the shells, because it is an acidic pH. Yeah. But efficiency will not be as great as of hydrochloric acid, but still it will work with that. Yeah. So, that is why mechanism and what way the technology works becomes more important rather than how to use it or where to use it. Yeah. If you know the mechanism, you can find a solution. So, now this is related to biofilm, which was previously confused as biopolymer. If you will see any other substance, if you spill a tea here, you will see some form of film formation happens. Yeah, because of milk and phenolics or whatever. Yeah. If you leave it like that, after some time it will become slimy. Yeah, slimy because microorganisms they grow. Yeah, and then if you leave for longer time, layer of microorganism, slime, then microorganism, then slime, it forms a biofilm. Yeah. It's easy to clean it on the surface. When it is inside the tube, becomes very difficult. You would have seen lot of tubes, they have microalgae inside. Yeah, microalgae or bacterial things they grow. Now, the problem is how do we get rid of this? So, these plasma species, they oxidize this organic matter also inactivate the microorganisms and get rid of biofilms. That means you replace antibiotic for usage. You will see some more some studies are present there. Directly or indirectly, they all are related to how do we reduce the wastage. Yeah, it's not only to keep the food microbiologically safe, you need to keep your hands also safe when you are going to handle it. Yeah, that is also important. It's not only the food is nicely processed, there is no microorganisms in a milk, you have milk bottle kept into your refrigerator, and then you make a tea and you just keep it outside for 2 hours, then put it back into the refrigerator. So how you handle the food, that is equally important than how the food is processed. Does it make sense? Now I will go through some of the study of plasma activated water and observe some of those inactivation curves. Yeah, within 1 hour 5 to 6 logs gone for E. coli. Same thing here, within 30 minutes gone compared to natural air. Yeah, this is all because of inactivity. I will go quite quick on these ones, because this is just you have to get the essence where it can be used, and this is not completely theoretical one to see what is it. And if you have any question, have it and we can ask. If you have any question related to any of the graphs, there will be multiple graphs, and then you can come up with your questions. Yeah. Now E. coli, how it gets inactivated? Oxidation. Listeria innocua, how it gets inactivated? Staphylococcus aureus, how it gets inactivated? Aeromonas, this is basically, Aeromonas is not mainly from food, but this is skin infection. So plasma is also used for washing of hands as opposed to alcohol washing. Look into Journal of Internal Medicines and Health Hygiene and Healthcare Hygienes. A lot of plasma systems are generated where they can be used for washing of hands. Plasma is also used in lot in a cancer research. I have 3 PhDs who are working only related to cancer research to kill those cancerous cells, yeah, through reactive species. Salmonella petrificans, Pseudomonas fluorescens. So this is all how it is inactivated. Yeah, simple mechanism, oxidation of outer membrane. Yeah, then all these reactive species, irrespective of the source, you get radical species. Then you have your proteolysis, that is breakdown of the protein, Maillard reaction, oxidation, disruption of the cells, and these are all the mechanism which leads to inactivation. But crux of that is oxidation. Yeah. So that is why oxidation and lipolysis. Now all this proteolysis or oxidation or lipid layer, if you know the outer layer of bacteria is what? Membrane made up of peptidoglycans. Glycan is a carbohydrate, peptido is a protein. Yeah. So surface oxidation happens and leads to pores, and because of pores those plasma species goes in and oxidize the cellular compound. Very similar how the insects they get killed. They breathe in oxidized air, it oxidizes the internal organs, end of story. Yeah. So that is the way how it operates. But the concentration required to kill microorganism is much lower compared to oxidize a protein. Yeah. So that comes D value. If you look into the literature, just find it out, D value of carotenoids and D value of bacteria. D value of bacteria at any given temperature is in seconds and minutes. Yeah. Whereas D value of carotenoids, if you want to get rid of 90% of vitamin C, how long you have to boil it at 72 degrees Celsius? Yeah. If you have to boil it, you have to boil it at least 4 to 5 hours to get rid of 90% of vitamin C from orange juice. Yeah. Whereas you yourself mentioned the pasteurization temperature is 15 seconds or a minute. Yeah. So there will be degradation, but it won't be at the level what is required to, the level required for microbial activation is much lower compared to oxidation of nutrients. Yeah. So there is always a trade off. Now this is some of the examples related to cold plasma. 16 kV, 70, 80 kV, 5 and 10 minutes, shelf life at 4, 8 and minus 20. Simple stuff. This is variac to change the voltage, and having a tray in between here is kept samples inside and out. Yeah. Lab, and how it's been gone to the commercial one is scaled up operations. You can see it in coming slide. Plasma activated water, this is a small ungutted sardines, whole fish dipped into a plasma activated water, yeah, and to see the microbial analysis. So what it does is a washing effect, so that whatever either you do it in water or you do it in plasma activated water, along with the ultrasonics. Next one is going to be an ultrasonic. Why ultrasound? Because it helps in absorption of plasma activated water on a fish surfaces. Now if you see apple and strawberry, or no, leave it strawberry, apple and cauliflower, yeah. Apple is having smooth skin, yeah. So microorganisms are easy to clean. When it comes to strawberry or it comes to cabbage, where the surface is very undulated, you will see a lot of grooves are there where the microorganisms will be sitting in, yeah. And if you dip it in the water, every portion of that crop will not have a will be accessible to microorganism, because they sits in some cracks or some small grooves, yeah. Whereas when you use the same thing with the ultrasonic, it allows it to go at the remotest place to do the cleaning effect, yeah. Same thing in a vessel. If vessel is having sharp edges, you will see everything deposits at the edge, yeah, because that is inaccessible. To make it accessible, combination of plasma with ultrasonics, yeah. So remember, it is done not because the technologies are there, yeah. At my place, we have developed a number of different systems, and ourselves we have around 18 different type of ultrasonic system, yeah. Some you will see here, and some are even non-existent. Those are designed in collaboration with the guys who manufacture. The people are ready to manufacture, to make an equipment for you, provided you tell them what you want, yeah. Our tendency has become to get an equipment to make use of it, yeah. Lot of places equipments are made. If you do not agree, just go to any of those small scale processors. They do make lot of different systems themselves which suits their purpose. I will give you an example. How many of you know a place called Theni near Coimbatore? Yeah. There was a place person was putting in your tur dal, Kajanus Kajan, that's Arhar in north, and there is tur dal, right? Filling in 100 kg and lifting a bag and putting into the weighing balance, yeah. Too much of energy was going, he was getting tired. And one of the place I saw, he just made an indentation on the ground, yeah, and kept the weighing balance exactly the level of the ground. He just drags it, put it there and moves it, yeah, right. There is no automation, nothing required, just a small modification reduces the energy consumption, yeah. So that is the way how you learn from different places. In order to propose any solution, first you need to see real life scenario. Real life scenario is not in papers, not in books, yeah. It is very similar. You cannot read a book and learn how to swim, yeah. You cannot read a book and learn how to drive. You have to do real experience how to drive it, yeah. So that same way, when you see the problem, then automatically you will get a solution. And I can bet you already have that much of scientific acumen and scientific knowledge, you can solve lot of simple problems, yeah. Every problem is not related to ultrasonics or so. Now this is now slightly complicated. And don't worry about this all peaks and all. This looks like electrocardiograph, yeah. Now once you have reactive species, how do we measure those reactive species, yeah. Simple way is to measure hydrogen peroxide by using a specific spectrophotometric method, nitrates, nitrite by using a spectrophotometric method for using Grease mechanism. That is what is easy to measure. But all these transient compounds are difficult to estimate. For that you need to have ICP OES, that is you have to have all the emissions absorbed, yeah. That means you get the spectra of the gas which is coming out, and then after that spectra is added to NIST library to find what corresponds to what. If suppose absorption is at 237 nanometer, that means it is related to oxygen species. So, these are the wavelength, yeah. And what is electromagnetic spectrum? Order of the light 380 to 780 is visible light, yeah. What is the order of the light? Vib, violet, indigo, blue, G, green, Y, O, orange, O, orange, R, red, yeah. More than that it becomes infrared, yeah. Near infrared, mid infrared, far infrared, and less than 400 ultraviolet, yeah. That is why the plasma beam what you saw was bit violet in color, yeah. And within that violet color which you cannot see from the eye, that can only be seen based on the spectra, yeah. So, this is not a rocket science spectra, it is the same spectrophotometer which is used to measure the spectra of the gas, and once you get the spectra of the peak, each peak represent certain species of electron or neutron. So, that is all what to do with the plasma, yeah. Okay. And this characterization is done at only one time with an equipment, and it becomes further complicated to measure these species when you are dealing with the food product, yeah. Is it clear? And simple measurement by using micromolar, this was done by using a spectrophotometric method for hydrogen peroxide, nitrite, nitrates, yeah. And to see at what treatment time how much concentration hydrogen peroxide happens. So, there is always a concentration and how it is left at different treatment conditions, yeah. So, there is no specific trend in all the gases, and these two nitrate, nitrites, they are always interconvertible, yeah. So, whatever available method is to see even 10 minutes of treatment time to create a plasma activated water, we can have that, yeah. So, this is just simple analytical method. I can bet you the moment you go quantification of plasma species into the literature, you will come out exhausted, yeah. You will not get a clear answer. To measure it, you just need to get the spectrum, yeah, with the spectrophotometer, the small spectro, not the spectrophotometer, but you get it, you know, that is a visible or invisible region. In that spectrophotometer big box, there is nothing, there is a cuvette place, yeah. You put a cuvette, there is a light source and light goes through it, whatever is absorbed, absorption what you measure, yeah. It works based on what principle? Beer-Lambert law, that means absorption has to be related to certain wavelength. Phenolics, fallen steel collateral, if it is blue, your wavelength region is around 600, reddish at a specific wavelength. Based on that you measure the compound, that means any phenolic compound, any bioactive compound, you add a chemical which only binds to a specific compound and gives the color, something like phenolphthalein indicator for titration, yeah. It changes color based on the pH. So, all the analytical systems are developed based on how you establish it. So, this is the what way the plasma species are characterized. And then once you have this spectra, to find which peak is related to what ion, for that there is National Institute of Science and Technology, this is online library available, you load that and you will get all those informations, yeah. So, that is about formation. Now, so far in plasma we have not talked about the frequencies also. The normal electricity what we get is around 50 kilohertz or 50 hertz. It is hertz, 50 hertz, right. But when we design a plasma system, there also frequency plays an important role, because if you are using the frequency in a kilohertz zone, plasma species formed are different. Crux of the matter is You have the air, you have to break air into ions, and those ions causes microbial inactivation or different application. That is all with the plasma system. Yeah. Details, devil lies in details. The more deeper you go, more mess it creates. Yeah. So in order to understand, important thing is to see how plasma is created and what way it happens. So this is again, it shows how it inactivates microorganism. This is the same graph and same system, right. Now coming back with the again voltage control treatment unit, this is mackerel fillet packed at non-MAP, that is not modified atmosphere. That means we are not adding any gas to it, it is normal air, and then this image is only seen when you see it in a dark violet color. Now you know why it is seen as a violet color. Yeah, you cannot see from the eye because it is invisible region. Yeah. Now if you will see number of microorganisms, Enterobacteriaceae, Pseudomonas, hydrogen sulfide producing bacteria. Now no pathogenic work is carried out on those type of species, because in that case this system has to be in category 3 lab. Yeah, that means having a different regulations. So you can see one second in the previous slide here, within this time we can get up to 3 to 4 log. Yeah, at least in a fish which is expected to have at least 2 log. So what is the difference? 7.5 to maximum you get is 1 log. Yeah, where is the rest of the species? Why it is not inactivating? So inactivation in a model solution versus real food system is chlorine also can give you 5 log if it is in a water or in a saline solution. The moment you have the same microorganism in Brussels sprout, broccoli or any other vegetable, the microbial reduction will be less, because some of those species will be absorbed by the nutrient to oxidize. Yeah. So this is all shows we can get significant amount of microbial reduction up to 90%, so that we can improve its shelf life. Now this is the question related to lipid oxidation, that is T bars. When I said we had a challenge with the T bars, because mackerel is also pink color, when you do T bars it also gives a pink color solution, right. So you get confused whether the pink color is coming from the meat or a food, or pink color is because of the reaction. That these are the some of the limitation of methods when we use it for estimation. Yeah. So whenever you estimate anything, you have to write what method was used for estimation. Protein by BCA method, protein by Lowry method, or protein by Zeldal method. Protein by Zeldal method will always show you higher level of protein. Yeah, because it will measure amino acids, peptides, everything as a protein. Yeah. Whereas BCA method will only estimate the whole protein or protein which contains tryptophan. Yeah. Estimation of method and type of method used is very important to report. Yeah. So as you can see, lipid oxidation was observed, but with no specific trend. Yeah. That is just output of this. You cannot draw any conclusion from here. And if you go with your standard deviation, all was something like this. So there was no significant differences. Yeah. So it becomes, we stopped it there. Then the question was further to see whether this oxidation is happening because of hydrolytic rancidity or because of oxidative rancidity. These two important role it plays. So what could be the selection of gases when it comes to plasma? If you have high fat product which contains large amount of fat, you have to refrain using too much of oxygen. Yeah, oxidative radicals. So if you investigate every free radical, every plasma gases on individual food compound, and to see what is the composition of product, based on that you can design a package, what type of gas to flush in and what plasma condition to use. Yeah. Okay. Because if you are not having a controlled conditions, say for example today relative humidity is around 90%, plasma will give you a type of inactivation, and next day you are repeating the experiment and it is rainy. Yeah, almost 99 to 100% relative humidity. You understand relative humidity, right? What is it?
the amount of water content, ratio of amount of water content is observed to maximum amount of water content.
Correct. That is why it's very important to know about the relative humidity definition correctly. Maximum amount of water what air can hold and how much it is holding, yeah. Because sometimes, you know, when I said this somewhere, so, oh, relative humidity is 100%, say, oh, then where is the oxygen, how we will breathe, yeah. So definition is important, amount of moisture it can hold. So if humidity changes, the moisture in the air changes and then the plasma species will change, yeah. Same 30 kV on dry day versus wet, not the dry day of 2nd October, yeah. Dry day of low humidity versus high humidity, results are not reproducible. Everything else remains the same. Reason is plasma chemistry. That is why the air has to be used of the same combination. Same thing happens for spray drying. Majority of the pharmaceutical application, nitrogen is used for spray drying. If air is used, air has to have the specific moisture, otherwise you are altering the air. Now when we use a nitrogen, the problem of nitration of protein happens, yeah. So there are a lot of, every solution comes up with the problem, yeah. When you like a rose, you have to take care of the thorns as well, yeah. So that's what it shows, and during storage, you can see lipid oxidation was increasing in non-thermal plasma, non-treatment, that is control, whereas on other plasma there was no specific trend, yeah. So that means all these oxidations were naturally happening without, if you do not use a plasma, then also oxidation happens, because we are not removing the occluded oxygen, the oxygen which is present into the structure. That is another reason why we do the blanching. When we do blanching, volume is reduced, turgidity is gone, because oxygen which is trapped into the biomass is gone, yeah. So that's now surface decontamination. Now if you will see in this particular one, this was a model food solution, yeah, of same sardines, because this sardine was to be used to make blind side sardines. Initial water wash, ultrasonic wash. Ultrasound did not inactivate, but it does the washing effect. On surface there is a microorganisms, if you wash it with hand, you reduce it, yeah, some reduction. But if you use an ultrasound, it will also remove from the small gaps and fissures what are there on the surface. Now in this case what you see here, when you have a distilled water, DW, yeah, you get almost 6 to 7 log reduction straight away in 10 minutes, yeah. The moment this is not a fish one, this is a model solution. If you add 5% of fish gelatin, inactivation takes bit longer to achieve the same, yeah. What does it shows? If you have to reduce 1 log reduction 4 to 3, this one is done in 10 minutes, in 1 or 2 minutes or 5 minutes. This one is done at 1.5, this one 5 hours, and this may be around more than that, yeah. That means model solution inactivation is easier, whereas when you are talking about the food matrix, inactivation is preserved because of two reasons. One, protective effect given to microorganism because of food macromolecule, and usage of that particular oxidizing agent in oxidizing those biomass or that macromolecule, yeah. Make sense? So if you see the key, ultrasonic washing, plasma activated water in model solution compared to fish, yeah. So that defines what you see is model solution 5 log in 2 minutes may not be applicable when it comes to real food solution. UV light treatment of distilled water, quick to get 5 log. If water is having lot of organic matter, it takes longer, because UV will be consumed by the organic matter to be oxidized, yeah. So same thing comes for biofilm. If biofilm is formed and plasma water is added in, how you can get rid of that biofilm. Yeah. Just look at this airborne acoustics. I will come back when we deal with ultrasonic systems here. Same plasma activated water, you put it on the surface, it will take time to penetrate into biofilm. Yeah. But is there any way we can increase the migration of that water into biofilm? By using ultrasonics. As you can see, airborne and microwave, no film. Normal washing, cold plasma, ultrasonic alone, no reduction, combination reduced. Yeah. Technologies are combined based on what is our objective. Yeah. So, this is the one which can get rid of biofilms along with it. Yeah. Now, this is the way looks how the control confocal is just dye is added, is absorbed by a specific part of the bacterial cells which imparts a color. The moment we get atmospheric cold plasma, see you would have seen lot of different terminology, cold plasma, atmospheric cold plasma, PJ plasma, that is plasma jet. Yeah. So, all these are just acronym, but to distinguish what is the mechanism there. Yeah. And then direct treatment, and then you can see reactive species and what happens to normal bacteria and what happens to reactive species or inactivation of microorganisms. Yeah. So, do not worry about this one, just trying to show how the things operate, and the intention is not to see going into the microbiology part of it, but just to have understanding how these inactivation processes they do work. Now, similar one, biofilm made up of E. coli, control ultrasonic, plasma, plasma, plasma. So, the moment we combine it, we get rid of biofilm. In some cases, when we are using microorganisms as a source of protein, we need that type of biomass, that is biofilm is required. In case of E. coli, that is a dangerous one because it will keep giving you the contamination. Suppose you have a conveyor belt, you are loading a fish on this and it keeps moving, yeah, to recover it, and over time the biofilm will develop on that conveyor. So, as process continues, this will add microbial contamination to your product. So, you need to get rid of the source of microorganism. Yeah. So, I am not going into the metabolic activity. So, it is just to go deeper into it, what causing microorganisms to inactivate. Now, if you will see this particular video, this is also a simple plasma system, and keep watching this particular video. Yeah. What this experiment shows, the two bread kept into a plastic container, closed container, in one there is a plasma, in one there is no plasma. Yeah. What does it shows? In 7 days, if bread is left in normal environment, microorganisms they attack the product. Where are they present? In the air. Yeah. Whereas, if you use a plasma, you are not treating the product, what you are doing is decontaminating the air. Yeah. As simple as that. And then this is all this normal fan, and this is the plasma what generates. Yeah. Now, learning from this, this particular system was designed, though it was for dried grains, where grains are kept here, and there inside is a conveyor belt, and there is a plasma species, and grains comes out here for decontamination. This you will understand by using this particular mechanism. Yeah. Now, this will, or this is having an industrial application. Direct treatment of plasma with the fish or food product will go through a regulation. Yeah. And I am pretty confident approval for food application is quite challenging to achieve. Whereas, in this case, this type of box was designed based on the previous experiment what you saw. Here you will see this is, how many of you have seen cold storage? Yeah. Or even this air conditioning room, what is happening? There is no outside air is mixing, only a small amount of outside air is mixed into the cold air. Same cold air is sucked and thrown back to the room. Yeah. So, that means if there is a virus in the room or microorganisms are there, if same microorganisms is circulated through the air. Yeah. although food-borne microorganisms are not aerial borne, but they move along with the dust, along with the suspended particle. So, same cold air, when it goes through the heat exchanger, this is the heat exchanger, these are the plasma devices which are added in, air goes through this and inactivates the microorganisms and you get a fresh air inside. Yeah. Further complication, what you see here is another automatic sensor is added to see when the microorganism starts growing. There also it was not a rocket invention, simple system was, I will just show you. If you have microbiological, how many of you have done the microbiological plating? Very good. I was very good microbiological plater because I used to adjust with the hand and whatever was target microorganism, apart from that everything else grows. Yeah. So, when you have microbiological plate and if you do pour plate method for inoculation, so how long it takes for you to count the microorganisms? Minimum 18 hours to 24 hours depending upon the bacteria. Yeah. Till the time you have a countable colonies. Yeah. Now, think this is the growth diagram of any microorganism, right? What is this phase? Lag phase. This one or exponential phase, and this is a stationary phase. Yeah. Same thing applies when you make a yogurt, you take always from the exponential phase. Yeah. Not from the dead phase, not from the this lag phase. So, this one is normally 1 to 2 hours max to max, in a media it is even shorter. Yeah. Now, if we have a optical sensor on top of this, yeah, and the plate one is very specialized media, yeah, if it sucks the air at a regular interval, throws onto the plate, yeah, if suppose this McConkey agar is used for what? McConkey. Sorry, Mike.
For coliform count.
For E. coli, right?
Oh, yes.
So, E. coli for McConkey agar is there, even I was not very sure, but I was thinking it is a E. coli or coliform. That's thanks for confirming and everybody will remember now. So, if it is for E. coli, if you have McConkey agar, then Listeria won't grow on it, right? It is only coli bacteria will grow. And if that one is spread onto that particular sensor, onto that plate, plate starts modifying, yeah, bacteria starts in growth, and the sensor will pick the changes on the surface of the plate that will lead to the signal that bacteria is about to grow or about to start contamination, then plasma can be controlled. Then you can call it as AI or machine learning and system in place. Yeah. So, that is why all this data logger are added in, yeah, and that is the way we can create contamination. This is what the type of storage system can be used for putting a fish, fresh produce or any other material where you are trying to eliminate the cross contamination. Is it clear? Yeah. Now, these type of systems, and then you have temperature control, you want to keep it 4 degree, 2 degrees, 5 degrees, 6 degrees or room temperature for different product. Yeah. So, this is, and where are the applications? Distribution system. How many of the cold room you have seen is disinfected? How many times you have disinfected your refrigerator? Never. You only clean it, right? So, that is why the refrigerator where you have a fresh produce and a meat, the product shelf life is very low, where the refrigerator where there is only vegetables, the shelf life of vegetable is longer. Yeah. So, that is why in all the supermarkets meat is separate, dairy is separate, fruit and vegetable is separate, but in home refrigerator that is not the case, yeah, because of same cross contamination. That means plasma can inactivate, refrigeration system is like this. Now, this is the way it is known as innovation system, yeah, the same plasma, and this type of plasma system has been installed in multiple number of airports, not only for oxidizing, removing the microorganisms, but also for to oxidize the contaminations in the air. Yeah. Now, we talked about number of different technologies. The question, some of you did ask a question, what technology to use for what. Yeah. So this becomes very, very important, what technology to select. First, your intention is only to kill vegetative cells, yeah, E. coli in juices or so. Model solution, whether it's a heat or non-thermal, you get more than 5 log. You have already seen that, yeah. When it comes to liquid food, we can get up to 5 log. Anything which can achieve 5 log is a non-thermal technology. We already discussed that, right? That means if you can jump 5 meter, you can jump 2 meters or 3 meters. Thermal, high pressure, pulse electric, ozone, ultrasonic, light based technology. But the moment it comes to solid food, achieving even 99% reduction, that is 2 log, is achieving a gold medal, yeah. Look into any of the research study published or any other report where you can get more than 1 log for chlorine washing, yeah. So this is one of the difference, yeah. Now, if vegetative cells cannot be inactivated, then you need to modify the technology what you are using it, yeah. That's one. You achieved it, then you have to determine the process efficiency, how efficient it is in real food situation, what is the impact on your nutrients, cytotoxicity, bioactivity, biological activity, then you establish this technology to be used for that purpose. If it inactivates the vegetative cell, second question comes, are you able to inactivate spores, yeah. Yes, do the commercial validity, end of story, yeah. If spores are not inactivated, then you go for combination. Example, other day we talked about high pressure plus heat, Clostridium, yeah. Then you go for commercial validation to see the impact on shelf life, nutrition, other things, yeah. So integration of any technology in a system has to answer certain set of questions before it can be adopted or can be used at commercial level. Now, any questions so far? Yes, Mike. Yeah.
Sir, as you mentioned about pour plate method, that it takes 18 to 24 hours for counting the microbial counts. Is there any new technology which reduces the time, or in which we count just in?
Yeah. Now, this is one of the biggest debate on between microbiologists and chemists, right? And even I end up in lot of argument with microbiologists in this case, yeah. I have even went into extent, if I get some infection, yeah, doctor says, oh, come back after 48 hours to determine what type of bacterial infection I have before he gives me a medicine, yeah. In clinical microbiology, the systems are already in place, and that is one of the most interesting area. For example, if something spoils, yeah, if suppose fish spoils, what happens? Gives certain smell, yeah. Whereas the same bacteria, or if apple spoils, there are some other bacteria. Each and every microorganisms are associated with specific type of product, yeah. So if you do the volatile profile, that is getting the volatile profile of a spoiled product. You spoil a product, create your library of volatile profile, yeah, and then if a product is spoiled, you take the same extract, dump into the into this MALDI-TOF. Do you know MALDI-TOF? Or untargeted metabolomics, yeah. Matrix assisted laser diffraction ionography, time of flight, yeah. I'll put it this way, coffee contains how many flavors, how many phytochemicals? More than 800, yeah. How many we know it? 30, 40, some are not known. So that is called untargeted. So same way, when microorganisms attack on a food product, it forms certain bioactives, certain metabolites, yeah. If we have that metabolite, group of metabolites identified in chemical processes, if a food product is spoiled, you take that water extract, put it in volatile profile, that means yes, this is happening because of E. coli, yeah. That's one way. Second one is direct cell counting, yeah. Taking extract and putting in it on and counting the type of cell it is counting, that means by using imaging processes, yeah. but everything is compared with plate count, but plate count is not the one which is most appropriate one, because if you see Rourkela is one city, yeah, and Bhubaneswar is also one city, yeah. Comparing two cities, one bacteria can form one colony, yeah, 100 bacteria can form one colony, yeah, but each of the 100 can form 100 different colonies, yeah. So, colony forming unit is counted, not number of bacteria are counted, yeah. So, that is why individual counting the cells and determination is one of the way to do it, or to go for chemical profiling method, or there is a quicker one is flow cytometry. Have you heard about flow cytometry? The images what you saw with the confocal, that goes along with the flow cytometry. That means flow cytometry, you put a dye to a specific, okay, take a fish, get an extract or saline solution of it, add a specific dye, dye will stain the peptidoglycan layer, like what you do gram staining, you have done it in the school, and then you put into a flow cytometer, it will show you the microbial population, yeah. If you heat it, see it, the dye is not retained by peptidoglycan layer because cell is already killed, cell death, yeah. So, you will not get exact population, getting exact population to compare with these methods. So, flow cytometry data cannot be compared with colony forming, individual bacteria cannot be compared with colony forming unit, because you are not measuring number of cells, yeah. Did I answer your question? Any other question? So far we have covered all about plasma technologies and how we can integrate that one into the system. The reason of covering plasma technology, because you have seen the plasma system, now you have fair idea how deep you can go, yeah, or how far you can go, yeah. So, we will we'll start with the technologies related to the processing using ultrasonics, which majority of you are aware of, yeah. Or ingredient, but same thing also applies for food and other applications, yeah. Because when we are talking about circularity, there is no starting point of a circle and a closing point of circle, yeah. So, everything is interlinked or interrelated, yeah. So, so far what we have covered is all about the biorefinery aspect, how to establish a biorefinery, and that is what you have your assignment as. If you remember establishing a biorefinery for seafood, you had what is the biomass, what is the composition, what are the end product you obtain, what are the side stream you obtain, and from side stream what is it rich in, and based on that you come up with the product. So that one I'll just have a quick recap later on when we go through the technology. You can have a look at it. Yeah. So that's one, and we did discuss about the plasma, and previously we used certain examples of high pressure processing, mainly for stabilization of biomass as well as improving the shelf life of the product. Yeah. Now we'll go through, for example, with the cavitation technologies, that is cavitation based on formation of cavities. These cavities can be formed because of water, that's hydrodynamic cavitation, or because of ultrasonics. So they do have both processing application as well as extraction application. So this is again the specific part of your novel technologies application. One itself is related to germination and drying. Yeah. If we use what we need for germination of anything, for any grain germination we need only moisture. We don't need a soil, we don't need a fertilizer. So if you have a technique which can absorb the water faster, so it will germinate faster. Yeah. So ultrasonics having one such application. Second one is drying. So even sonics treatment can leave the micropores on the surface, which also can improve for drying, as well as you have seen some example of airborne acoustic, how that can also be used for drying purposes. They are just representative examples. So we have post harvesting operations, food preservation, microbial inactivation either by sonication or thermosonication, and leading to improvement in shelf life of liquid, semi-solid and solid food products. So that is again food preservation. Nutritional aspect, so when we use cavitation, we have a capacity to preserve the nutrients because it does not cause much of degradation of nutrients. Extraction technologies, it's used extensively. Now it comes here for allergens. So whatever allergens are, it is also useful in modifying the functionality of the protein, that altering the properties of protein. Hence it can also reduce the allergenicity or increase the allergenicity. Yeah. So ultrasound is not always for inactivation but also for activation of microorganisms. Peptide production, same way, enzyme plus ultrasound or ultrasound followed by enzyme. So that is all for peptides. So this is all related to nutritional aspect, related to processing as well as extraction side. Functionality, ultrasound assisted crystallization of fats and sugars. Ultrasound is one of the technique which can control the crystal size and type of crystals. Mixing and homogenization, that means ultrasonic, if you have ultrasound equipment or access to it, take water and add oil to it and give ultrasound treatment, you will see perfect emulsion formed. That is again for mixing and homogenization. So other one is valorization. So extraction of high value ingredient, wastewater treatment, that means again breaking down of dissolved solids and improving reduction in BOD and COD. So if we use cavitation along with ozone or with the plasma, we also can achieve advanced oxidation processes, that is cavitation will help in diffusion of those ionic species into the matrix and enhances the distribution. So this can be used not only for processing but also for preservation and valorization. So all these applications can be for cavitation, and there are number of different review papers and literature is full of different applications of ultrasonics. Yeah. And there are already 2 dedicated journals itself on ultrasound. One is ultrasound sonochemistry, other one is ultrasonics. Yeah. So you can see the amount of application and science related to sonics. So that's if you look at the different application, as far as I am aware, there are more than 50 to 60 or close to 100 applications are there, but 50 are almost used in different scales for different application across agri-food sector. Yeah. Some examples will quote it what are related to biorefinery aspect. Now when it comes to ultrasonics, what are the key parameters related to that? This is very important to understand. I don't want it to, how many have seen ultrasound working, a noise of ultrasound? You can see this one. So first and foremost important one is ultrasonic frequency. Yeah. So sound operates based on frequency. What I am talking is also based on frequency. It moves based on compression and rarefaction. Liquid sound is different, metal sound is different, vacuum is different. Second one related to processing is your amplitude. It is the amplitude which is the guiding force for ultrasound. Ultrasound power, ultrasound intensity and ultrasonic power density. So, all those things, if you will see, this is ultrasonic probe, yeah, and this is normal water. How we play it. If you will see, if you add a small amount of powder to it, and the moment you give a blast, just observe the video, yeah, and that is ultrasonic probe which emits the cavitating bubble. And what you will observe is all these bubbles, these are all cavitating bubbles, yeah. So, if you are using a spoon or anything agitated, it will take ages to mix it together, yeah. So, this is a clear cut example of just the mixing. But here again, if you have a water and oil, it will create milky color texture, milky color liquid, that becomes exactly as an emulsion, yeah. So, all of you know about electromagnetic spectrum. Light, this, infrared, microwave, audible, and then different megahertz region. So, where all ultrasonic is used? Ultrasonic is used for both diagnostic purposes as well as processing application, depending upon the frequency. So, ultrasonic equipment, whatever is sold in a market and whatever is available, they operate only on a specific frequency. What you can change is only the amplitude, ultrasound power and ultrasonic intensity, yeah. So, amplitude is distance between two nodes or antinodes, yeah. That is again you have seen the classical Bell experiment, you hit it and then it start moving. So, if one is operating on a specific frequency and if you bring the another one of exactly the same frequency and they will go in sync, yeah. That is the way all the communication and everything is carried out, yeah. Now, this particular transducer is made up of piezoelectric transducer, yeah. How many of you know about piezoelectricity? We did discuss about piezoelectricity as a quartz, yeah. Can you give the mic to me? Yeah? One second. Yeah, go on.
Compressing or stressing a piezoelectric material, we get there is a change in the electric field of the resistance of the material.
Correct. So, if you press a material, their positive negative current separates and then you can get electric current. The simplest one which is used extensively is the quartz. Every watch is having a quartz in it, yeah. That is a piezoelectric property. So, when you apply a pressure, you can generate electric current. Same way, when you generate electric current, you can when you put electric current, it generates the pressure. So, that is piezoelectric transducer. If you see ultrasound, it says piezoelectric transducer. That is property what is used in these type of transducers. Similar properties are also there in biological materials. So, if you have any of the plants, if you will see, they also contain piezoelectric properties. Simple experiment is when pea plant grows, if you put one bamboo stick, one green plant and one metal rod, and you will see the node of that particular pea will go to a specific direction, yeah, how it senses. And these biomass, even chitosan also contains piezoelectric properties. Any material which can have separation of with application of pressure, it separates the current so that you can generate the electric current, and that can be used for making future batteries or different pressure system, yeah. One of the latest example of piezoelectric properties for making pacemaker batteries. Lot of patients, they have pacemaker is installed and needs a battery. So that particular battery is replaced with a piezoelectric material, so that heartbeat which gives the pressure, which will activate, which will keep that particular pacemaker in business by providing sufficient electricity. So these are the some of those advancements happening even in lot of institutions in India. There are lot of experiments and protocols are ongoing how we can take electricity from a growing plant. When plant grows, there is also ability to get electrical current out of it. Yeah. So the purpose is plus and minus current like that. So this is again difference between two crusts is your amplitude. So when it comes to equipment, how many of you have seen ultrasonic equipment? You have seen it, yeah? So have you seen a bath system or like a bath?
Sir, without bath system.
You've seen the probe system?
Yeah.
Okay, this type, yeah?
Yeah.
Okay. Now, can you give it to her, yeah?
Bath system, sir.
Bath system, yeah? So, what is the difference between a probe system and a bath system? We'll go through that one, and if you have any question, feel free to ask. So, getting the clarity is very, very important, yeah? So, what we can change is basically amplitude, power, intensity and acoustic power density, yeah? So, unit of amplitude is going to be in unit of distance, that is micrometer. Amplitude cannot be anything. Power, watt, yeah? Ultrasonic intensity, watt per centimeter square, that is dependent upon the radiating surface. If probe is of 1 centimeter diameter, area of the probe will be 3.14 centimeter square, yeah, because pi r square, I am wrong, 1 centimeter, that means 1 upon 2, that means 0.5 centimeter, yeah, that is pi r square. What I am saying is if radius is 1 centimeter, yeah, radius is 1 centimeter, area of the radiating probe is pi r square, that is 3.14 centimeter square, right. So, if that radiating surface and watt is measured, watt upon the radiating surface becomes ultrasonic intensity, yeah. And acoustic energy density is if you are treating 100 ml of water, so what you know, watt per ml will become acoustic energy density or acoustic power density. So this was the unit was first established by ourselves. We did that with the what way we calculate ultrasonic intensity. There are multiple other ways of calculating ultrasonic intensity. Having ultrasonic bath, you put your aluminum foil, you have seen those experiments. Amount of aluminum which is removed from that foil, that could be one way. Second one is the measurement of hydrogen peroxide. When we do sonication, it breaks the water into OH and H plus, and that gives you idea of how much hydrogen peroxide is formed. But all are dependent upon power, because ultrasonic power is the easiest way to estimate by using caloric method. Sorry. So that is all the basic about ultrasonic. I will come back to you how do we measure ultrasonic power. Now another thing related to this ultrasonic system is, if you did your experiment for 100 ml water and then now you want to use the same condition for 200 ml, it won't work, yeah, because acoustic energy density distribution is changed, yeah. That is one of the limiting factor when it comes to scale up. So when you have to scale up, you have to accordingly balance out acoustic energy or power density going in. What I mean is, if you are putting 10 watts for 5 minutes in 100 ml, so if you increase 100 ml to 1 liter, same amount of power should be gone to 1 liter. So you have to increase the time, yeah. So that's the way your scale up operations are carried out. That is applicable to every technology, whether it's plasma or plasma, pulse electric field, any technology, whatever you deal with, you have to work accordingly. So how we can calculate ultrasonic power? What is given on the machine is the machine rating, yeah. This television or this projector is working on 2000 watt is a maximum it can draw out the power, or a fan with the 600 watt is a maximum power, but if you are using at lower speed, you are only taking 50 watt, 100 watt or 200 watt, yeah. So that is nothing to do with ultrasonic equipment power. So those values which are written on the publication and things, they are not correct precisely. That is one of the reason you cannot reproduce some of the work which is already there and based on the similar conditions. Yeah. So if you do something with ultrasonic bath and you do something with the probe, both cannot be replicated. Reason is simple. In ultrasonic bath, anyway, I will show it to you in next slide. Yeah. So this is again electromagnetic spectrum. Yeah. Before going into the application, I will say how we calculate a power. Suppose this is your beaker, yeah, and you have a probe in it. You put 100 ml of water, yeah. 100 ml of water, what will be the specific heat of water? 4.18 kilojoules per degree Celsius per kg, correct? Specific heat of water, amount of heat required, that is joules, to heat a water or per kg of water by 1 degree Celsius, right, that is 14.5 to 15.5. So this is a specific heat. So when you start when you start the ultrasonic system, temperature automatically increases. Yeah. When temperature increases, if you measure the temperature, that is rate of change of temperature over time, that gives you dT by dt. Yeah. That means if temperature increases over time, yeah, so you measure the slope of that particular line. Slope of this one will be, suppose this is your temperature and this is your time. Yeah. When you take a slope of this, so dt means delta t. Yeah. And those who are from physics or maths background, they do understand that. And this is time, right? You measure the slope of this. So calculation of power will become, what is the equation of power? M Cp delta T by delta T. Yeah. Slope, you get it from here. Specific heat is here. Mass is 100. Yeah. So you get your power. This is calorimetric method for measuring the power of ultrasonic system. Now the question comes in the moment if I add a juice to it, yeah, or a food matrix. We only power represent through distilled water because specific heat is remain relatively the same, yeah, and you all know the specific heat changes with the temperature, with the pressure, with the compounded material. Specific heat of orange juice plus pineapple juice, it will be difficult to determine. Yeah. So we just get a representative of getting calorimetric method. Yeah. So then this way you get your watt, then you know about the diameter of your probe, makes an ultrasonic intensity. Whereas when it comes to bath system, the radiating surface is bigger. So power will not be the same what you get it from 200 watt probe system versus 200 watt bath system, because ultrasonic probe is attached to a flat surface and whole surface is radiating. So intensity is reduced. Yeah. That is why comparisons are not easy to make between a bath versus probe. You might come across lot of publication or lot of papers, it says the ultrasonic probe is better than ultrasonic bath. It is correct, but it is not level playing field, because one is not able to deliver the power what other is able to deliver. Yeah. You carry out the race for 200 meters, you ask Usman Bolt to run and you ask a 5-year-old kid to run. Yeah. Both are humans. Yeah. You can't compare it. So the similar way. Now when you look at the frequency sweep, infrasonic, acoustics, human hearing zone is over here. Yeah. And ultrasonic, anything above 20 kilohertz is classified as ultrasonics. So majority of the processing application are between 20 to 100 kilohertz. Some applications are there at this zone, but anything above 5 is goes as a diagnostic one. So this will be called as high frequency low power ultrasound. This is low frequency high power. So majority of these applications are related to making sensors, diagnostic purpose, detection of tumors. How many of you have gone through ultrasonic in hospital? You put a sensor it tickles, it sends sound waves and it determines the structure. Yeah. So, all these one lower frequency, cleaning, cavitation, higher power. Yeah. Low power, imaging, sensors, microscopy. So, this is the way ultrasonic system can work. Now, when ultrasound operates, you get both mechanical effect and a physical effect, both are same anyway, mechanical and chemical effects. Mechanical effect can be used for crystallization, degassing. In almost every chemistry lab you will see ultrasound, and people who are working on analytical chemistry, they use ultrasound for removing gas, degassing, because cavitating bubble gases are removed. Yeah. So, that is degassing or foam destruction. You will see one or two examples here. Extraction we already discussed, mixing, homogenization, brining and tenderization, freezing, encapsulation, drying, tempering. Just now I gave you an example where you have oil and a water. If you give ultrasonic treatment or even a blast, it will give you a perfect emulsion. Yeah. Same way, if you have a starch solution, you give an ultrasonic treatment, it will form an emulsion. So, that means this is one way of doing encapsulation. Encapsulation is covering of any ingredient with filler material, that is bioactive, covered with other, what do you call the outer layer of encapsulation? Protein or starch or any of those material can be used for encapsulation. Now, if you do it at a microscopic level, microencapsulation, yeah. Capsules what we consume as a medicine, they are also in capsules, they are also encapsulated outer covering. Yeah. So, it is not to bug down with the terminology. Other one is a chemical or a biochemical effect. Bactericidal action because of production of free radicals, effluent treatment, that is again reduction of BOD and COD, modification of living cells. Previously I said you can use ultrasonic to activate the microorganism as well as inactivate the microorganism depending upon the condition we use. Yeah. Very similar to a temperature. If you use 70 plus temperature, you will kill a microorganism. Whereas if you use 30, 35, 40 degree temperature, you increase the microbial growth. Yeah. Similar principle applies here also. Alteration of enzyme activity, because you can modify the protein properties, hence you can modify enzyme activities. Sterilization, like thermosonication, heat plus ultrasound will cause sterilization effect. Yeah. So, this is all how ultrasonic can be used. I have not listed other application like soil treatment, soil remediation, heavy metals, coating. Ultrasonic is used extensively for non-food applications also. Yeah. Now, if you look at the processing equipment, we already discussed just now about bath type and probe or horn type. Yeah. That will need a medium to deliver ultrasound. Yeah. What way we are communicating, though it is not ultrasound, but it is air which is we are using it as a medium. Yeah. That goes as airborne acoustics. Now, if you see, this is the bath arrangement where the transducer is placed at the bottom, yeah, or transducer are based at sides depending upon the system. Even two bath system you cannot compare, because that depends upon how the transducer is configured. Yeah. Probe system, very same transducer is injected directly into liquid. Yeah. And this is also a transducer. This is a flat transducer, you can add it into any bath system. This is not a probe, but a transducer which operates at 400 or 600 kilohertz, and this frequency is very good when we extract oil from olive oil, pomace or any of those biomasses. Yeah. And there is no relationship between frequency and extraction. What works at 25 may not work at 45 or may not work at 100. What works at 100 may not work at 600. So, it is always its ultrasonic is case dependent. Yeah. So, that is why the huge amount of applications keeps coming in for ultrasound. Even solid mercury or liquid mercury was prepared by institute in Israel by using ultrasonic. Yeah. Nanocarbons, carbon nanotubes activated, carbon nanotubes were prepared using ultrasonic. the black color powder which is formed. There is a lot of different application. Now this is also ultrasonic bath, but at larger capacity. Yeah. Now these are different types of probe system. How many type of probe system you have seen? You have seen single probe, yeah? So, sorry.
Sir, I have seen single probe, but different size, different length.
So different size and length, that means they deliver different amplitude level. In order to know the amplitude level, you have to go to the manufacturers. They only can tell you what is the amplitude of that machine. Measurement can be done, but again you need a precise tools to do the micrometer measurement, right? So that means different diameter, that dictates how intense you are delivering the ultrasonics to it. Yeah, that is important here. Now when it comes to ultrasonic, as time progresses, you will see the holes in the probe at the bottom of it. Yeah, that's called pitting after certain size. That's what is written here. Repeated usage of ultrasound will, initially when you buy it, it's perfectly smooth. As you operate, it starts removing the titanium. Most of the probes are made up of titanium. So that removes the cause pittings on the surface. Easier way to remove it is to grind it to make it smooth. Yeah. And other one is you get a replacement tip. You just replace a tip, you attach to it. Yeah. Similar way boosters are attached to ultrasonic probes. Now if you remove it and similar frequency, tuned frequency, you can get it for clinical application. If suppose you want to study impact of ultrasound on 96 well plate, you can have 96 probes, so that you can measure a probe to prove the concept. Then this is your probe system, and this is batch up to 1 liter or so. And this is a very similar process where you have a vessel where you have liquid coming in. Yeah. This is for controlling the temperature. Liquid goes in and products comes out. Yeah. So how we scale this one? Suppose you need only 5 minutes treatment time for 100 for 1 liter, and this vessel is of 1 liter. You control the flow rate so that whatever liquid passes through this gets 5 minutes treatment. That's the easiest way to scale up, or a simplest way. Or else you need to know the acoustic energy density, and based on that we can scale it up. Yeah. Now if you will see here, suitability limited to liquid or food immersed in liquid medium, because you need to have a liquid there. And one question comes all the time is, if we are using ultrasonic all the time, the pitting, that means leaching of material, comes into the liquid food. Yeah. We carried out that experiment extensively and spoiled almost 10 to 15 different types of probe to see amount of metal which leaches. So the amount of metal which leaches is exactly the same way we keep using the pressure cooker, and you see slowly the metal, whatever vessel you are using for cooking, it removes the metal. Yeah. Similar thing happens here, and after the prolonged application also, the residues in the samples are much lower than the standard of that particular water. Now if you have read about ultrasonic, everywhere you will see the temperature of 3000 Kelvin, or pressure of 400 or this much. 3000 Kelvin temperature is of sun's surface. Yeah. So you are using an ultrasonic, so how you can create such a high temperature? What is the basis of that high pressure and that high temperature? The basis of that high pressure and temperature is indirect measurement, because if you need that much amount of temperature to break the water, based on that, when it breaks the bond between H2O, H2O is not is not an easy molecule to break into H plus and OH. It needs huge amount of energy in terms of temperature. Yeah. So based on that temperature, these type of energy is calculated. Yeah. That's why you'll see like 3000 Kelvin or how many megapascals, couple of thousands of pressures are reported, and these are all measured and established how much it can generate. Yeah. If you see your own classical textbook of flame, what is outside temperature, what is inside temperature, light blue is the complete burning of fuel, temperature map of any candle when you burn. Yeah. Yeah, any question? Yeah. Okay. Now this comes airborne acoustic. Yeah. This is ultrasonic plate which delivers ultrasonic at higher frequency, 25, 50 or 55 depending upon how it has been fabricated, and it delivers the cavitation in this particular manner. Yeah. So as you can see, the gap is 40 centimeter, and one or two examples you are seen in with the plasma on airborne acoustic treatment. So this is the transducer, piezoelectric ceramics to transfer the electric current into the pressure transducer, and it gives ultrasonic waves through this. Yeah. This is airborne acoustic, completely non-contact. Even these systems can be used as a non-contact if you have it in your beaker, or if you are having it in your pouches or in surfaces. Now when we use ultrasonic system, lot of places you would have seen people, they use round bottom flask. Yeah, that is completely wrong, because ultrasound will deflect. It always have to have a flat bottom. Yeah, if you are putting in a bath system, it's a round bottom, so how much surface is exposed to it. Yeah, so there are some fundamentals of using ultrasonics in chemistry. The certain books are there that is very important to read before you start using ultrasonic for any experiment. This is one system configuration based only on ultrasonics, A. Second, now you will be happy, ultrasound plus heat. You are working on thermosonication, right? So what you are doing?
Sir, giving moderate temperature with ultrasound.
Moderate temperature.
Yeah, yes, sir. 40 kilohertz of frequency, and the temperature is about 50 to 60.
Okay. So when you use heat along with ultrasound, it becomes thermosonication. If you use ultrasound plus pressure in a pressurized chamber, it becomes manosonication. That is combination of pressure and sonication. If you use all three together, it becomes mano-thermosonication. Very simple. Yeah. Based on combination, these nomenclatures are in existence. So if you use 72 degrees Celsius with ultrasound, you can kill the spores. Yeah. That becomes, ultrasound helps in causing the pores on the spores, and when the temperature is used, it helps in killing that particular spore. Now it becomes slightly complicated. Now we go with this. Yeah. Now if you see this particular system, sometimes we may have to use multiple frequency system. Over here, this is 45 kilohertz bath system, and these are two probes both of 20 kilohertz, that is dual frequency system. Yeah. And this is a commercial level pilot scale system was developed, and you will see the application of this. This is your tank to supply cold water, cold brine to this particular vessel, and having ultrasonic bath as well as probe system. Second combination, as you can see, this is a domestic microwave, yeah, which was modified to do the treatment. Now looking at this, you don't have to modify the microwave. This is not that easy to do it. Our trained engineer has to do that, where to cut that particular microwave. And as you can see, ultrasonic probe is going into that particular microwave. But this ultrasonic probe is not made up of metal. This is custom built probe, which can be also classified as a plastic probe, yeah, which is dipped into microwave to get the combination of both probe system and microwave together. So we had our own IP on this, which was lost because there was a French system which designed much better system, having ultrasonic at the bottom and microwave coming both into the steel vessels. How many of you have added the spoon into a microwave? What will happen? It will cause spark. Yeah, because in microwave we can put liquid or solid food, but the moment metal goes in, it starts causing spark. But in this case, microwave goes into the steel vessel and ultrasound is at the bottom. The trick here is ultrasonic moves with a specific frequency. That's why, how many of you seen microwave, domestic microwave? Yeah, there is a rotating plate. Yeah, if plate is not rotating, there will be burning at a specific points. Yeah, this just because microwave also travels in a wave. Yeah, it is electromagnetic wave. So if it is moving, so that it is having uniform heating. So in this case, there is a Teflon stirrer which moves, which guides the microwave, does not allow metal to come in contact with that particular microwave. Yeah. And when we use metal and ionic current into microwave, we can also generate microwave plasma. Yeah. Now this is this is all to do with, oh sorry, this is permanent one. Yeah. So this is basically, if you look at ultrasound and supercritical, this is supercritical system, which would, I was trying to remove this circle. So this one is having a vessel. This is a controller where the CO2 is pressurized, passed through the bottom of this tank, sample is loaded inside, and then liquid CO2 goes out, pressure is released, and you get your product out. Yeah. Now the design was done when the CO2 is in liquid state to use ultrasonic to disrupt the matrix. Yeah. You won't have seen much in the literature, and you won't be seeing it because we only got it designed recently, and that's still looking at it. The purpose here was something like you have a hexane, you add into the matrix, so oxalate 6 hours to get the lipid, right? But if you disrupt the matrix along with the hexane, recovery can be done in a shorter time. Same thing when you are using a supercritical CO2, it will take longer to extract all the oil from the biomass. But if you disrupt the biomass along with the liquid CO2, so it will have a better extractability. So for that reason, ultrasonic system was attached along with this. Yeah. So this will be synergistic aspect of both supercritical as well as ultrasonics to be exploited. Yeah. All these combinations are not done just because like a Lego, the kids, they play Lego, they made a truck out of it, they made a tractor out of it. It's not added just like that, they are added for a specific reason. I will show you some of the examples in coming slide. Now we discuss about the plasma jet or steam. When you throw a plasma, it moves in a form of air. Yeah. So in order to direct that onto the surface, if suppose this is a full of plasma species, if you remember I was saying if you have to inactivate the biofilm, that species should enter into that particular matrix to kill it. Yeah. So there airborne acoustic helps in suppressing those reactive species into the matrix by causing the pores on the surface. Yeah. So majority of the combinations are based on the logic and what works, what does not work. Steam treatment, if you do the steam washing, you would have seen the steam jet washing, they are pointed. But if your steam jet is in a flat layer and then you suppress the steam, then you will have a uniform cleaning just to improve performance. Yeah. So it will have lot of different voices, and when we talk about cavitating bubble, look at this. This is a cavitating bubble and how it implodes, and this is what is known as microjet and explosion. Yeah. Because of that you get all those reactions. When you read ultrasonic, says formation of microjet, acoustic streaming and all those things. Yeah. This is all to do with cavitating bubbles. Yeah. So this is the bubble, look at it again, vacuum, and this is imploding, and because it's implosion it is forming microjet. Yeah. Now here this is microjet formation, and these are now if you will see here, I am not going to turn on the sound, 10 micrometer, these are cavitating bubbles. Yeah. These are at different now if you want to measure these cavitating bubbles, yeah, diameter of these bubbles can be measured and can see the reactions. How can you measure that? What do you need? Very high speed camera. Yeah. If you have high speed camera, you can track the bubbles to see how the cavitating bubbles operate. And this is the same 20 kilohertz ultrasonic system, yeah, 20 kilohertz frequency system operated at using 125 micrometer probe. This is your different percentages of amplitude, and accordingly there will be power. So as you can see, this is all acoustic streaming, microjet, and single principle is cavitating bubble, yeah. Cavitation is single principle, rest all are effects of it, yeah. And it makes really very loud noise, yeah. And when you are using ultrasonic, suppose 750 watt, and if you want to really use 750 watt, take that ultrasonic probe, put it into wood, yeah, any hard surface like wood or anything. So ultrasound will always try to vibrate at the same amplitude. If it doesn't get that much of place to vibrate, it will use the full power, and if it's not able to use full power, it will gone, yeah, that is overloaded. So for that reason boosters are used. So if you put a water, it will make loud noise. Put 2% sodium alginate solution, voice will be completely low. It's just because this sound is not because of ultrasound, but because of those cavitating bubbles which are forming, yeah. So that's why ultrasound does not make any sound, because if it makes sound, we can hear it, yeah. If you turn on ultrasonic bath without water, I know, never, don't try it because it will spoil the system. It won't make any noise. The moment you put a water, it gives the sound. That is all because of bubbles colliding with different matrices. Now, if you look at this particular system, anything when you have direct field pressure, this is simple compression and rarefaction, yeah. So nothing happens. Second way is stable cavitation, that is cavitating bubbles are formed, yeah, and they are just vibrating, yeah, that is stable cavitation pressure. Transient cavitation, when bubble is formed and it is vibrating, exploding, imploding. So there are two types of cavitation, as stable cavitation and transient cavitation. That means in this case, sound wave is going, particle is happily standing there, just simple. In these cases, micro streaming happens, that means bubbles are formed and they are moving here and there, yeah, like what you saw it in the video, all these bubbles whichever are moving, that is micro streaming. Because of that, it dislodges the particles or disrupts the matrices. Another aspect, advanced one goes in to make shock waves. Somebody is working on a shock waves, that is one of the way also to produce electric shock waves or sound shock waves, yeah. And jet, formation of jet, and this causes disruption of matrices, yeah. So the single principle is cavitation. This cavitation could be stable cavitation, transient cavitation, and these cavitating pressures can be calculated and can be estimated by using a specific type of sensors, yeah. As you can see from here itself, all these cavitating bubbles when they are moving and how they are forming jetting system. And this was done with the normal camera in a beaker, clear beaker, to get those videos, yeah. And the purpose was to measure those bubbles. Then we realized we need very high speed camera, which is quite expensive, to slow it down and then to measure the bubble and what is the fate of that particular bubble, yeah. Now this is what I already discussed briefly, right. Now power MCP dt by dt, the same delta T, this is differential, because when you are measuring temperature over time with different amplitudes, you get the slope of it, yeah. Once you get the slope of it, you get this value, yeah. Specific heat you know, mass you know, you get power in terms of watt, yeah. Ultrasonic intensity, power upon surface area of radiating surface, yeah. That is pi r square. To make it more fancier, you do pi d square by 4, because diameter is easy to measure, whereas radius is difficult, yeah. Acoustic energy density, power upon volume. Yeah. I'll ask you a question. Suppose there is a tree trunk. How can you determine the radius of a tree? By using a vernier caliper. Yeah. How can you measure a diameter of a tree? By putting a scale or something. What is the easiest way of doing it?
Sir, we will measure a small area and then we will integrate the whole.
Mathematician, good. Any other simple technique? How I can measure? I have studied only agriculture. This was the question which was asked by my own undergrad UG teacher when we had a module on agriculture engineering. Yeah. There was a simple way. There you use a basic math. Take a thread, tie it around 4. You measure the parameter. That way you can calculate a radius. Yeah. Make sense? Because the question was measure the diameter of a sugar cane. Oh, I need a vernier caliper. That's one. For measuring the distance from this wall to this wall, you need a measuring tape. But a technique called pacing. Yeah. So you know what is the distance between your normal walking. Or if your shoe is around 10 number, 9 inches or 31 centimeter, 30 centimeter, shoe after the shoe, you can get rough estimation, right? So everywhere you will not have those sophisticated tools to measure it. Yeah. So always try to see first one is a simple solution, then to go for complicated one. Yeah. That's correct. You can use mathematically and all. Yeah. But how do we measure it? How do we know whether it's 12 o'clock or 6 o'clock based on ratio? If you see the old historical way of measuring the time. Yeah. So all those techniques are very useful in a practical application. Yeah. That does not mean we should not know the advanced method, but basic method knowledge is equally important. Yeah. So that was a simple one. Just take a thread, measure it around. You know the parameter. Parameter formula is pi d. Yeah. 2 pi r. That is pi d is parameter. So pi d is equal to the length of the wire or thread, and then you get your diameter. You don't have to have a vernier caliper. You don't have to cut anything to do all those integration and disintegration. So other complicated. This is simple method of measuring it. Other ones, acoustic emission, acoustic pressure distribution, cavitational efficiency, bubble collapse intensity, bubble temperature, number of cavitating bubbles. These are other ways of measuring energy and acoustic distribution of bubbles. Yeah. So these are all number of different techniques. Simple one, for as far as processing is concerned, these are sufficient enough. Yeah. Any questions so far? Yeah. Yes. Your name is Kavya.
So my doubt is on multi-frequency bath with probe type, sir. Whether the product is led to process simultaneously or in batch types.
No, both together.
Both together.
Yeah.
Whether it will be effective, sir.
It was effective. That's why it was done.
So, because I have came across some literatures like the cavitation created by one piezoelectric transducer affects the other piezoelectric transducer.
Completely agree with you, because if you are having two transducer together,
yes, sir,
they both will be destroyed. Yeah?
Yeah.
So when these type of things are used, you have to use it. Suppose you have two probes. Yeah.
Yes,
it has to be like what you saw, the flat probe which was kept 400 frequency. They cannot be kept like this. They have to be kept like this. Okay. Yeah. So that is the important aspect of it. You read it correctly. Okay. Because if you put both, sound pressure comes from here, sound pressure comes from here, two different frequencies, it will destroy the transducer. So it has to be in a perfect alignment with that. So here it was done basically the length of was different, and then to combine both the frequency, and in that particular bath system you can have a transducer from the side, not from the bottom. Yeah.
Okay.
So that's why transducer configuration becomes an important part of it. Yeah. Same thing when we did for microwave extraction. You can use the water for extraction perfectly fine. The moment you put hexane and ethanol, it creates a vapor, and any spark can cause fire. Yeah. So usage also depends upon what is the solvent. So you need to know the dielectric property of a liquid when we use it. Same thing for pulse electric field. If there are particles, it will cause a spark, or you cannot have air bubbles in a pulse electric field. Yeah. I cannot cover all the technologies, but these are some of the limitations for each and every technology. So it has to be used based on understanding their proper working, yeah, and proper mechanism. Then only we should think of applying. Yeah. Now microbial inactivation. Now this is the viable cell. When give ultrasound treatment, it causes pores on the surface. Yeah. This cell is not dead. Yeah. It causes pores and it can improve its performance so that substrate can go into the cell faster and reactant can come out faster. It only damages when pore size is slightly bigger, so that all the cytoplasmic material comes out of it. Yeah. So there are studies where you do a calibrated pores because of ultrasonics, so that you can transmit DNA, cDNA or specific drugs into the system. As you can see from here, this is much clearer. This is favorite bacteria of dairy technologists, Lactobacillus sakei. Yeah. This is the normal cell. Yeah. You give ultrasound, you can see all the small pits. Yeah. This is at controlled energy delivered. Now this is also a pores. Yeah. And then you extend the energy, then you get leakage of cellular content. Yeah. Is something like aqua or acupuncture. Yeah. You make a holes in it, so the substrate can go faster into it and end product can come out of the cell. Yeah. So reaction will be faster. So ultrasonic can also be used, especially a bath system can be used for improving the fermentation rates. Second reason is, when you add a microbial culture, you mix it. Yeah. But if you add a microbial culture, you give an ultrasonic, low power ultrasonic within that zone, it will distribute the cells widely. So it can improve the performance. Same thing happens for microalgal cells. If you want to increase the growth, you can give ultrasonic controlled power so that you have a better growth into the suspension. These are all open-ended questions. You investigate it, we'll read it. Yeah. So that's pretty much open area. Yeah. Now this is TEM. We carried out the work on yeast to see how yeast can cause the pore, why yeast fermentation is improved. And when TEM was done, what was found, both the bacteria were working very well, but vacuoles disappeared. You know, every cell they have vacuoles. Vacuoles disappeared. Then there was another question the person who was doing, what is the role of vacuoles in the cell? Otherwise we will stop it there. Otherwise we go into the fundamental to mechanism to other aspect. Yeah. So that's the way the question starts with the simple problem and you get more and more complicated. That's why you create more scientific knowledge. Yeah. Otherwise it is a cook and look. Then in order to do that, we also wanted to see, is there any genetic modification happening in a cell, or why it is utilizing better nutrients. So there again, I am just stopping it there because otherwise I will enter into utilization of nutrient, how it is modifying the system. Remember, only two different mechanisms, one causing the pores onto the surface so that it can have a better reaction, substrate transfer. Second one is dislodgement of that particle, even distribution, like what you saw in first slide when you had a small black powder added with ultrasonic probe. Yeah. If you increase the energy into it, it will kill the cells. Yeah. So same thing like when you acupuncture, that therapy, you put a holes, it improves some of the problems of the body, but if you make a big hole in a body, what will happen? Yeah. So the same thing works here. Now ultrasonics, the same mechanism what we have already covered, comparing with thermal. Yeah. So thermosonication can inactivate these microorganisms or to inactivate spores. Yeah. By using heat and ultrasound together. Airborne acoustics. Now, what is the application of airborne acoustics? Now, we are a bit digressing from your biocircularity. Since I mentioned about the technology, it is important to conclude it for what it can be used. Non-contact microbial inactivation or process intensification. Take this example. Yeah. How many of you have boiled milk? It comes up, right? What do you do? Yeah. Same thing ultrasound does. Because if you have big vat, you can't do it. Yeah. So this sound pressure goes on, it suppresses the foam, so that you don't have to add anti-foaming agent. When you are extracting the protein, it will create foam and it loses the product. So you can use it as anti-foaming agent. Yeah. This transducer, there are a couple of patents are there for design of those transducers, and these are industrial scale. Now, if you see this example, we all drink cola beverages. Yeah. And this cola, not all, I don't drink much anyway. So when you fill a carbonated beverages into a bottle, product loss happens because of bubbles. So when it is filling in, airborne acoustic is kicked in, suppresses the foam. Yeah. If in a day you have 2% losses, it's a huge cost. So defoaming application. Yeah. So our purpose was not to use defoaming. Our purpose was mainly to use it for inactivation purposes and to use airborne acoustic along with the drying system. So this is a radiating plate and its focus point is 10 centimeter square, yeah, at a specific area. So this was not a small system, it's a big system. As you can see, distance is 405 millimeter, that is nearly 40 centimeter. And this is the distribution of acoustic power within this particular chamber. So maximum intensity in the center and how it is reduced along with it. So this can be measured by using hydrophone, by using normal sensor for measuring the sound intensity. Purpose: diagnostic probe, plasma jet filling in the plasma species and causing microbial inactivation. Yeah. Now when it was used, remember I was talking about high value spices and condiments, we don't use high temperature, just because if you use high temperature they won't have those flavors. So they have to be dried at low temperature. So when we use airborne acoustic, it also helps in drying and also reduction in microbial load. But the timeline what you see is quite significant, yeah, 60 or 90 minutes. So if this is combined, whether it's a black pepper or tapioca starch or any starch coming from any biomass or a protein, for drying, when we use this, we get significant reduction to a certain level, that is 1 log or so. And then if we combine with the plasma, this time can be reduced A. If we use it at 40-degree temperature, it will dry off faster. Yeah, that's why when we are going through the seaweed, you would have seen ultrasound plus airborne acoustics. Yeah, now we are covering about the technology. Yeah, so this is again percentage degradation phenolic compound. When was done with the black pepper, no changes. Yeah, because it's airborne. Starch, no changes in rapid visco analysis, that is with rheological properties were not changed and we don't want it to get it changed. Yeah. So these are just demonstrating different application. Some of the material is already there in print media. If you are interested in any specific thing, I'll be happy enough to send it to you. Now, where this particular airborne acoustic and plasma system was used. Now suppose this is the surface for biofilm, stainless steel, plasma water alone, thermal plasma, activated water, that means two different type of plasma water, and airborne acoustics, that is the same system. Control and ultrasonic was able to dislodge not that much. Water, plasma, thermal plasma, like different mechanism, different gas chemistry, when they both are attached with ultrasonics, complete removal of biofilm. Yeah. So airborne acoustic, opening up the structure and plasma species causing inactivation. It is very similar. There are two people, one is breaking the door, other person is having a gun. You might have played lot of video games like that. Yeah, it is a similar thing. So then it's synergistic method. Yeah, okay. So that's the way it operates. So this will just simple how we can improve the efficacy of specific system. Same thing if you do not want to use a plasma activated water, even if you are using a chlorine, you can reduce the usage of chlorine by employing new technologies along with it. Yeah. So, this is only your confocal microscopy to show how the things are changed only with control and ultrasound, dislodgement and then inactivation. So, we are not going to go deeper into it, just when we talk about technology, it's my duty to show where it's been used. Yeah. Now, coming back to your ultrasonic bath system, meat curing, fish curing, any of those one, brined fish. What is the purpose of brined meat or brined fish or brined pickle? Biomass added into salt solution, right? Salt slowly goes into and moisture comes out, your osmotic dehydration. But the same osmotic dehydration is carried out in ultrasonic system, you have a better result. It will speed up the process. So, one normal method is, okay, it's becoming, one is normal dry salt. That means you have a product, you throw a salt on it, something like when a snail is walking around in a garden, you put small amount of salt, it dies. Yeah, that is basically it draws out its moisture. That's why salt can kill, to take the moisture out. Yeah. So, in a meat you rub a salt on it, and after rubbing a salt, if you vacuum pack it, put it into ultrasonic bath, salt will diffuse uniformly. Yeah. Other one is immersion. You have brine, you put fish, you put meat, you put pickle, like what we do pickling. Yeah. And then other one is brine injection. That means you have a tough matrix, you inject a brine into it and leave it, you get salt migrated into it. The main purpose of brining process is the salt enters into the matrix. Now, how this system was designed? One, when the brining is carried out at commercial level, all those things are kept at 4 degrees Celsius, so that microbial growth does not happen, another thing. So, in this case, this is the brine with the pump. The brine was passed through the heat exchanger to have 4 degree. Rather than having a big tank maintaining the 4 degree temperature in a tank, we can have a normal tank, circulate the brine through the heat exchanger and to have a chilled effect. Yeah. Correct in saying that you have individual air condition in the room or you have a centralized AC. So, which one will be more efficient? Centralized air conditioning system. Yeah. So, similar way, instead of having multiple tank, each tank is maintained for 4 degrees Celsius. Lot of heat losses are there because you have to have a metallic steel tank. Instead, normal tank and brine is circulated through heat exchanger. Yeah. And then brining. So, example over here, piece of a meat going into the system and brining time, that is ultrasound and normal sodium chloride content of the product. Yeah. And then how it happens, this is the interesting part, not all this one. This is the surface of a meat. Yeah. When we give ultrasonic treatment, if you get a proper SEM images, you will see surface becomes like this, porous. Yeah. These are not like a big pore, these are at micropores because of ultrasound. That is why we call it sonoporation, and these red dots are sodium chloride measured by using a SEM EDX. So, when we do the salting, salt only goes between the fibers of a food product, as you can see over here. This is muscle fibers and salt is in between these. Yeah. Whereas, when we do sonication treatment, there is a uniform distribution of salt. So, whether you are using a brining purposes or you are using as a solvent for extraction purposes, mechanism remains pretty much similar. Now, any questions so far? No? So, some more valorization example we will cover when we go through tomorrow for biopolymer or feed application. Now, I added this slide. Yeah. Majority of us, not only you, including us, we are at this zone. Yeah. Who can look into it? Yes, I can grow microalgae in a wastewater. Done. Paper published. End of story. Yeah. Whereas there are only few technologies which are commercial. A lot of you were asking question, what will be the cost of this? What will be it? Yeah. What technology transition? Yeah. Majority of the equipment which comes in a market, you don't, suppose you buy a domestic microwave, you don't change the frequency, you only operate based on whatever frequency is. So equipments are set to operate on a specific conditions, yeah. Fan, you don't change AC, DC, pulse duration and all those things, right. So once we know where we have to integrate it, so technology, research and development, demonstration, system development and mission operation and integration. So a lot of those type of systems, even in NIT Rourkela, you can see how those systems are designed, like automatic pickling machine, automatic dosa maker. What is the new technology there? Yeah, you've seen that. Automatic chapati maker, an automatic fryer for vada. You put a batter, it creates creates your vada and it moves to the oil and it turns and then you get the product, right. That is all technology development. You demonstrate it and then you integrate into the commercial line. It is not as easy as what is seen. So this is what I meant by technology readiness level. Basic principles observed, yeah. Then you prove the feasibility and then you develop accordingly. Like this morning, what's your name again, sorry? Kamaveshwari. So this morning she mentioned about when she used osmotic dehydration, lipid analysis was, lipid oxidation was less, yeah. So that is what basically what you have observed, yeah. Now you try to develop that, try to process in that line, that itself becomes a technology. It's not that, oh, it's just normal osmotic dehydration. No, it's not. Canning was discovered by Louis Pasteur way like almost 100 years ago, or yeah, nearly about 100 years ago. But still canning process is upgraded every time, yeah. So that's the way technology develops. Technology does not mean all those fancy probes, supercritical fluid extraction added here and there, yeah. What developed based on the need. You observe it and then you move forward. Same way piezoelectric transducer, piezoelectric properties of biological material was observed quite lately, and then that particular material now to run into make a biomaterial to replace quartz or to replace batteries. We don't have enough of metals to make a battery. So same thing as a solar plant, yeah. Solar system was not new, now you have EV cars, electrical cars. So that means it's been running. So that slowly development happens. But before things operate, you have to be in this zone. Majority of the food processing operation comes into this circle, yeah. So a bit how it works will be the commercial uptake of the process. Having those two things in mind is important while doing any processes, yeah. But remember, you cannot answer everything in one single experiment, yeah. So any questions so far? No? Actually this particular slide was for market uptake, value chain and how we integrate it, yeah. But remember, I will come back to this one again, right. So that is why it is important to know what we do, yeah. It is good to have those questions, yeah. I do get often these questions, whether this technology can be used in industry or what will be the cost of the technology. I have simple answer, take my phone out and this one. 1990, nobody was having phones, yeah. Like you all have iPhones. When we were studying, we had no phones, yeah. But now systems are there. Slowly systems are upgraded. So, but who should get the Nobel Prize for phone? Who is the first inventor? Graham Bell, yeah. Developed that phone, and everything is developed based on that, yeah. Same way your osmotic dehydration was done from centuries, yeah. But now you see where it can be used, yeah. So then you slowly nurture that one. You don't ignore. That's why observation becomes very important in any of the unit operations what you do, yeah. Okay, that's great. Thanks. Oof, I should have reduced my speed.